हेलो हेलो देख आवाज आ रही है यूट्यूब यूट्यूब अरे उसको दूसरा एक फोन दो ना यूट्यूब के लिए हेलो 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 आ रहा है ना आ रहा है ना आ रहा है ना चेक 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 चेक
विनीत आवारी हेलो 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 चलता उतना
sir. Good morning, sir.
गुड मॉर्निंग।
Yes, sir. सर आप ट्रायल लेने वाले थे? आप ट्राई कर सकते हैं सर, आप डायरेक्टली स्क्रीन शेयर कर सकते हैं वहाँ से।
ओके, ये अभी प्राइवेट मोड में हो रहा है या सबको दिख रहा है ये?
सर ये सबको दिखेगा सर, यूट्यूब पे भी जाएगा और जो हमारे
नहीं,
सेमिनार हॉल में पार्टिसिपेंट्स हैं उनके लिए भी।
अभी ये जो ट्रायल रन कर रहे हो ये सबको दिखने वाला है? मेरी आवाज ऑडिबल है?
जी सर, जी सर।
ओके, बिकॉज आई एम गेटिंग एन इको हियर।
सर हो रहा है सर शो, हम देख पा रहे हैं सर आपका प्रेजेंटेशन पूरा, जो टाइटल स्लाइड है। ओके।
ओके, so let me just check. So how now you can visualize is a full screen or split one?
सर फुल स्क्रीन पे रहेगा सर ये।
हाँ, ये अभी आपको जो विजुअलाइज हो रहा है फुल स्क्रीन विजुअलाइज हो रहा है ना?
हेलो।
तो आपको ये फुल स्क्रीन विजुअलाइज हो रहा है या स्प्लिट विंडो है? हेलो।
हेलो। सॉरी सर, थोड़ी आवाज़ नहीं आ रही थी। मैं बात करता हूँ सर। मैम
सो आपको ये अभी स्क्रीन जो दिख रही है ये पूरा फुल स्क्रीन स्प्लिट विंडो दिख रहा है? हेलो। कैन यू हियर मी?
हेलो। हेलो।
कैन यू हियर मी? मेरी आवाज आ रही है इसमें?
जी सर, आ रही सर आवाज़।
तो मुझे ये पूछना है कि अभी ये जो स्क्रीन आप देख रहे हो ये फुल स्क्रीन में प्रेजेंटेशन की स्लाइड दिख रही है या स्लाइड स्प्लिट विंडो में दिख रहा है आपको?
पूरी स्क्रीन दिख रही है सर आपकी।
ओके।
हेलो सर।
हाँ। यस।
जी सर, आप बोलिए सर, अभी मैं थोड़ा सेमिनार हॉल से बाहर आया एक्चुअली, तो वहाँ पे थोड़ा सा आवाज़ इको।
राइट, आई एम गेटिंग द इको एज वेल।
यस सर, वी आर वर्किंग ऑन दैट, वी विल रिजॉल्व इन 5 मिनट्स।
कैन आई चेंज द स्लाइड?
hello.
structure prediction for co-crystallization studies and its applications in formulations development and the use of SilicoXplorer in artificial intelligence driven drug delivery platform. Alone we can do little, but together we can do so much. That means अकेले हम बहुत थोड़ा कर सकते हैं, but हम साथ चले तो बहुत कुछ कर सकते हैं. So today we are gathered here to explore the involving frontiers of pharmaceutical research, artificial intelligence, driven drug discovery, a domain that is transforming global healthcare. But together we can do so much. To begin this event, I invite all the but हम साथ चले तो बहुत कुछ कर सकते हैं. Forward. So today we are gathered here worshipping the goddess Saraswati with Saraswati Vandana and lamp lighting. Research, artificial intelligence, driven drug discovery, a domain that is transforming global.
या कुन्देन्दु तुषारहारधवला या शुभ्रवस्त्रावृता या वीणावरदण्डमण्डितकरारध या श्वेतपर्णहारधवला या वक्रतुंड महाकाय सूर्यकोटि समप्रभ निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा. वक्रतुंड महाकाय सूर्यकोटि समप्रभ निर्विघ्नं कुरु मे देव सर्वकार्येषु सर्वदा. As Atithi Devo Bhava is our Indian culture, I invite all the guests to occupy their seat on the dais. Please occupy the seat on the dais. As Atithi Devo Bhava is our Indian culture, I invite. So our respected guest of honor, Dr. Sushil Rao sir, Director, Biocent Research Private Limited, Pune. Our today's chief guest, Dr. Praful Sabre sir, Professor, Department Science, IPA Nagpur. Our respected guest speaker, Dr. Muhammad Ataullah Islam sir, Director and Head of Science, SilicoScientia, Private Limited, Bengaluru, and Honorable Dr. Vinod Thakre sir, Principal, Nagpur College of Pharmacy, and Conference Conveyor, Professor Dr. Pramod Khedekar sir, Nagpur College of Pharmacy. Thank you all for illuminating this seminar with light of knowledge and innovation. May I request the organizing committee members to present floral bouquet to our respected dignitaries as a token of gratitude and respect. For this, I invite Dr. Vinod Thakre sir to give floral bouquet to Dr. Praful Sabre sir. May I request the organizing committee members to present floral bouquet to our respected dignitaries as a token of gratitude and respect. For this, I invite Dr. Vinod Thakre sir to give floral bouquet to Dr. Praful Sabre sir.
sir to welcome Dr. Sushil Raut sir with floral bouquet as a token of love and gratitude. With the sapling, as we change our floral bouquet with the sapling, sapling is permanent solution for the flowers rather than bouquet. Now I would like to invite Dr. Praveen Surse sir to welcome Dr. Muhammad Ataullah Islam sir. With the sapling, as we Dr. Praveen Surse sir. Permanent solution for the flowers rather than bouquet. Now I would like to invite Dr. Sachin Hiradev sir to welcome Dr. Vinod Thakre sir with sapling and token of gratitude and love. Dr. Praveen Surse sir. Now I would like to invite Dr. Sachin Hiradev sir to welcome Dr. Vinod Thakre sir with sapling and token of gratitude and love. Now I would like to invite Mrs. Namrata Mane Madam, HOD Nagpur College of Pharmacy to welcome Dr. Pramod Kedekar sir, Professor Nagpur College of Pharmacy. Now I would like to invite Thank you. Mrs. Namrata Mane to become a center of excellence in the field of pharmaceutical sciences to create versatile, competent pharmacists in the vision of Nagpur College of Pharmacy and under the MEGI group of institutions having the milestone set up since 2019. This is our first international seminar organized under the guidance of Principal Dr. Vinod Thakre sir and Khedekar sir. I invite Dr. Vinod Thakre sir to give small introduction about the journey of Nagpur College of Pharmacy and about today's event, that is International Seminar and Drug Discovery Training Program. Having the milestone set up since 2019, this is our first international seminar organized under the guidance
Thank you,
of
madam.
Principal Dr. Vinod Thakre sir and Khedekar sir. I invite Dr. Vinod Thakre
Good morning,
sir to give small introduction about the journey of Nagpur College of Pharmacy and about
distinguished
today's event, that
guests,
is
respected
International
speakers,
Seminar
industry
and Drug
experts,
Discovery Training Program.
faculty members, principals, dear researcher, and my dear student. Good morning to all of you. It gives me immense pleasure and pride to welcome you to this one-day international seminar and drug discovery training program on the theme crystal structure prediction for pre-crystallization studies and its application in formulation development and the use of an AI-driven drug discovery platform. This academic international seminar has been made drug discovery training program valuable support on the theme and collaboration of the Association of Pharmaceutical Teachers of Studies India and Maharashtra application in formulation development, Indian Pharmaceutical Association Nagpur Branch drug discovery platform. Dr. Baba Sahib, this Ambedkar Technological University Lonere has been made drug discovery training program valuable support on the theme and collaboration of express prediction for pre-crystallization studies India and Maharashtra for joining hand formulation development, Indian Pharmaceutical Association Nagpur Branch in pharmaceutical education and research platform. Dr. Baba Sahib, this Ambedkar proud privilege to extend heartfelt welcome valuable support on the theme chief guest and collaboration of Professor Judicious Nagpur Maharashtra for joining hand formulation development, Indian Pharmaceutical Association honor Nagpur Branch in pharmaceutical director and platform. Dr. Baba Sahib Research Private Limited privilege. I also heartfelt welcome valuable support on the theme chief guest and collaboration of Professor Judicious Nagpur Maharashtra for joining hand support and contribution guest of honor Nagpur Branch organization director and platform. Dr. Baba Sahib Research Private Limited privilege. I also heartfelt welcome valuable support on the theme chief guest and collaboration of Professor Judicious Nagpur Maharashtra for joining hand support and contribution guest of honor promoting strong research director and platform. And developing public research private limited journey of we are grateful for the Bengaluru award leadership and constant Professor I respect and acknowledge and shared support and contribution whose inspiring vision continues to shape our educational mission. I also extend my sincere gratitude to Honorable Sri Raviji Meghe sir, Secretary Sri Vidyarthi Sudhar Sangh, for his continuous support and motivation. We are equally thankful to Honorable Samirji Meghe sir, Treasurer and MLA of Hingna constituency, for his valuable administrative guidance. My sincere Honorable to Honorable Sri Raghavji Meghe sir, Trustee Secretary Sri Vidyarthi Sudhar Sangh, and for his continuous support and motivation, Chief Advisor. We are equally thankful for Honorable constant inspiration and commitment towards academic excellence and MLA of their leadership and blessings for his valuable administrative guidance behind the steady growth. Sincere achievement of to Honorable Sri Raghavji Meghe sir, since Secretary inception, for encouraged college has continuously for his considerable support and motivation, Chief Advisor, we are equally thankful for Honorable upgraded digital governing facilities, academic excellence, and nurture of dedicated and experienced team of faculty members. Valuable we have expanded our academic program. Honorable enhanced industry interaction. Encouraged research publication. Honorable patents and motivated Raghavji Meghe since Secretary co-curricular and extracurricular social professional activities. Our aim is not to create the graduate, but to nurture responsible pharmacists, innovators and future leaders of healthcare system. Keep silence. We are privileged to host a renowned expert from across the globe. Today, we extend a warm welcome to Dr. Amit Pant from Creighton University, Dr. Mahanti Munikoti, Mycoliver, Germany, Mr. Vikram Singh Suryavanshi, Dr. Mohammad Ataul Islam, Director and Head, SilicoScientia, Private Limited, Bangalore. He is basically from Bangladesh. Along with all session chair and resource person, your presence elevate the academic spirit of this program, and we are truly grateful for your willingness to share your expertise. We are living in the era where pharmaceutical science are rapidly transforming. Artificial intelligence, computational modeling, and data-driven approaches are redefining how we design molecules, predict properties, and develop safer and more effective medicine. The integration of crystal engineering with AI-based platform is accelerating research and opening new avenue for precision, formulation, and personalized therapy. For students and researchers, this seminar is a golden opportunity. Today, you will interact with experts, understand global research trends, and gain insight into technologies shaping the future of drug discovery. I encourage every participant to actively engage, ask questions, and absorb as much knowledge as possible. At our institution, we strongly believe that education must go beyond textbook. Our focus remains on skill development, industry readiness, research attitude, innovation, and ethical responsibility. We are continuously working towards achieving higher standards of accreditation, strengthening collaboration, and contributing meaningful to society. I wholeheartedly congratulate the organizing committee, faculty members, and student volunteers whose hard work has made this program a grand reality. Your dedication reflects our collective commitment to excellence. Once again, extend a warm welcome to one and all dignitaries, participants. I am confident that today's deliberations and training session will be highly enriching and inspiring. Thank you, one and all. I wish the seminar grand success. Thank you, thank you, thank you, one and all.
Thank you, sir, for enlightening the informative words. To continue with today's event, now I invite Dr. Swati Gaikwad, Madam, to give brief introduction about our today's guest of honor, Dr. Sushil Rao, sir.
A very good morning to one and all. Myself, Dr. Swati S. Gaikwad. I am an associate professor at Nagpur College of Pharmacy, and I take this opportunity to welcome all the respected dignitaries on the dais, distinguished guests, faculty members, and my dear delegates. It is my great pleasure and honor to introduce the guest of honor for today's auspicious occasion, Dr. Sushil Y. Rao. Dr. Sushil Y. Raut is a pharmaceutical scientist with over 16 years of academic, research and industrial experience, currently serving as a director at Bioscience Research Private Limited and honor to introduce his expertise spans medical and scientific writing, nanocarrier-based drug delivery, formulation development, analytical and bioanalytical studies, and clinical data interpretation. He has previously worked with leading organizations including Novartis, SIRO Clinpharm Private Limited, and Indigen Private Limited, and has also served in academic roles as assistant and associate professor. Dr. Rao has made significant scholarly contributions with 35 plus research publications, book chapters, and has an H-index of 16, focusing on nanomedicine, oral protein delivery, lipid-based carriers, and advanced drug delivery systems. He holds 4 patents related to nanostructure lipid carriers, engineered nanocarriers, polymeric encapsulation, and peptide conjugated liposomal formulation. His achievement includes the Praise Award for Research and Publication by MAH, Lady Tata Memorial Trust Research Grand Finalist Recognition, International Travel and Conference Grants, and Best Poster and Citation Award for Impactful Pharmaceutical Research. Overall, Dr. Raut's work integrate innovative drug delivery research, scientific communication, and translational pharmaceutical development, contributing meaningful to the academia, industry, and a global healthcare research. We are truly honored to have such an eminent personality among us today. May I now request our esteemed guest of honor, Dr. Sushil Y. Raut, to kindly address the gathering and share his valuable thoughts. Let us welcome him with a big round of applause.
Thank you, ma'am, for this wonderful introduction. Good morning, everyone. Still, Josh is not that much. Good morning, everyone. Yeah,
Good
that's
morning,
the
sir.
Josh. So, before proceeding to address this gathering, I would like to thanks to the organizing committee for organizing this wonderful event. So, all the dignitaries on the dais, faculty members, students, and all other who contributed to conduct this event, even at back end. So, thanks to everyone. As proceeding at this position as a director of Bioscience Research Private Limited, I started my journey as a student, turned faculty, turned researcher, and then entrepreneur. This journey was not easy. I am from the nearby village. You might be aware about Umred. Basically, I am from Umred, and there was no particular ambition when I started my journey as a pharmacist. I did my B.Pharm from IPA Varda, and some dream was shown by Dr. P.G. Avle sir, he is a Vice President of PCA, Pharmacy Council of India. And one of the personality who was my, who is my friend and companion person throughout my career, Mr. Dr. Vaibhav Uplanchiwar sir. When I started my journey, there was a little spark came when I was in B.Pharma, and I've gone for the M.Pharma and PhD. I got an opportunity to pursue my full-time PhD at Manipal, then postdoc from Taiwan. I was a hardcore researcher. I used to sleep in laboratory, and I learned lots of techniques on, actually I'm from pharmaceutics person. I developed various formulation, nano formulation. That time trend was there, nano formulations. I also started working on nano formulation, but I always used to think something different, out of box, and based on that, I developed certain platforms so that it can be helpful to the society. Very simple formulation I developed, that is, just now I talked with sir, that is patented technology for that car perfume. You might be aware about Ambipure, right? So I developed in third year I learned one principle, thixotropy, gel to soul to gel technology. So I thought when we are using certain marketed products, it is getting exhausted within a short span of period. So why we should not go for the product that gel, that fragrance can long lasting up to 6 to 1 year. And I use gel lock technology, and I developed the gel which can work on the principle of thixotropy. Simple principle which I learned in my third year, I actually adopted and got a patent for that. And that is a utility patent. Very simple, not more than 3 components. Not more than 3 components. So nowadays many people are doing the lots of research with the lots of complexities. But if socio-economic point of view, if you think the company don't want the very complex formulations, they require very simple formulation so that even a common person can buy it. So they don't want a complex product. So whenever you are developing certain formulation, you think over that. So today's event is organized in focus of artificial intelligence in drug discovery point of view. So artificial intelligence is the current need, and definitely as per the experts from the AI field like Sam Altman, Jensen Huang, then Bill Gates, then many other like Elon Musk, what they are predicting that in coming 5 years, all these existing technologies will be replaced with the AI. Even though currently we are using in support with the existing technology, but in future, if you think about robotics, there will be the lots of attritions in the job field, even though in pharmaceutical field. I'm not saying that you may be replaced, but if you are not going with the flow or across the flow, we can say, you will be replaced by the person who will be knowing the AI. Example, it's very simple example I'm giving, security guard. After next 5 years, in next 5 years, you will find robotics sitting in the security cabin. Okay, he will give access to people, he will trace the people. If anyone is forcing, he will even fight with that person and giving alarming system. So you will get it in 20,000 rupees, 20 to 50,000 rupees. So instead of paying money by your management to the security guards, it can be replaced with the robotics. And they even don't go for leave or many other issues. So that will be affordable also. But thing is that they will replace the job of securities. But new job may be getting created, so people will require to keep watch on those robotics. Okay, system controller and robotic controller system and many other analysts. So it doesn't mean that existing things are going to change. So traditionally, if you go drug discovery and development process, it was taking around 10 to 15 years. Serendipitous techniques. So it's, drug discovery was based on the trial and error basis. So they developed the minoxidil as a vasodilator for hypertension, but found that it applied for the hair transplant therapy. Okay, hair regeneration therapy. So it is accidental therapy. Like that, on trial and basis and serendipitous ways, we are getting the medicines, and nature is the base of all the medicines. But later on, when computational technique has been arrived, people started developing, started optimization, lead optimization and screening techniques with this computational technology in a very fast manner. And now, the you might be aware 2D, 3D QSAR, now 5D QSAR are there, and along with that, to get ease and precision, to do the to save the lots of time and improvisation in your precision and results, AI has been utilized along with the existing computational technologies. So that screening time is saved. So everything is most of the peoples don't want to get all the molecules directly into the laboratory. They are using the existing softwares. So simulation has been involved, AI has been involved, and with that precision, when the optimized molecule can later taken into the laboratory for the synthesis and screening, and then can be preclinical testing can be done, and then can be taken for the clinical use, after regulatory approval. So that's saving the time. Now you might be heard about remdesivir, how rapid they have developed that medicine. Okay, Moderna and Covaxin, how much faster way they have developed those vaccines. So that time has been reduced, that efforts has been reduced, and even the investment done by the companies has been reduced because of all these tools. That's why these are more benefited and utilized or adopted by the industries. Now, this is all about industry point of view, drug discovery point of view. So more details can be explained by the company people here. So what exactly, how you can go for molecular docking and other process. Now student point of view, I'm talking here, what, as I work in industry, what is industrial expectations? Most of the students and their parents are thinking that as my ward has taken admission for B Pharma or engineering or medical, they must get a job, right? You join this course to explore the pharmacy field, to get skilled in that and to get a job in this field. You are not taking this education taking this degree for any charity purpose, right? Your purpose is to get a job, to earn the money and to grow in this field, right? Many students are approaching me, and what they are expecting job first. Why company should give job to you when you are not skilled? That's why these type of events has been conducted by the institutions, so at least you can get exposure to the new skills. So company will not waste their money only on degree. Degree has no value until and unless you are not skilled. This is the primary need of any company. If you are lying on your resume that you are attending certain seminars, theoretical concepts are not useful. Practically you should be skilled. And if you are showing that practical skills and that is reflecting in your work, irrespective of your degree, company can promote you, can give you more hike as well. So there will be lots of chances. So what I am suggesting all the students, focus on skills. So with current technologies, you might be not aware. So always aware about technology. So what company is looking into the students? As I work in the industry, I work in close association with the HR and learning and development teams and with scientists. What they are expecting from you people, your psychometrics, your knowledge, your skills, your communication, your interest and your SWOT. If your psychometrics, you might have the team dynamics. Sometimes you may be independent player. You may have decision taking ability. Sometimes you are following others' decision. Skill is definitely, no one is replacing that skill. If you are skilled, even though other things you don't have, you can develop it. But if you have skill, no one will replace you. Knowledge, additional support to the skill, that knowledge will help you. Then communication, because you are communicating with the different peoples, your clients or your colleagues, your subordinates and even superior people. So your communication should be prospective to getting the business, to getting the research oriented way. And SWOT analysis. So you should know yourself. So whatever 6 points I told, psychometrics, knowledge, skill, communication, interest and SWOT. If you are working on these 6 things, no one will stop you from being lead the your job roles. Okay, so you start working on that, leverage your CV in this perspective. So there are lots of research and interdisciplinary things we are learning. So first thing, you should decide your goal. Your goal should be smart. You have to focus what exactly you would like to do, whether you have to go in computational drug discovery or AI based drug discovery or formulation scientist. Everyone have that unique interest. Okay, that's why I said interest is also important. Sorry. Oh yeah. So, so ultimately there is a thing that skill is very important, and no one can replace the skilled person. So try to leverage your skill and build your CV and explore the new things. So thanks to everyone for giving this wonderful opportunity to speak in front of you. Thanks one and all.
Thank you, sir. Artificial intelligence is not replacing pharmacists, that sir has told us. And it is true, it is empowering the human being. Now, I would like to invite Mrs. Hina Mehta, madam, to give introduction about today's chief guest, Dr. Praful Sabre, sir.
Good morning, everyone. On behalf of Nagpur College of Pharmacy, I am Mrs. Hina Mehta. It is my great privilege to introduce our distinguished guest, Dr. Praful M. Sabre, sir, professor in the Department of Pharmaceutical Sciences at RTM Nagpur University, Nagpur. With over 29 years of experience in teaching, research, and academic administration, Dr. Sabre, sir, has also served as the Director of Board of Examination and Evaluation at RTM Nagpur University. He has been a Central Council member of Pharmacy Council of India nominated by the University Grants Commission. I would like to invite Dr. Sabre sir to please guide us with his words. Thank you, sir.
Thank you, madam. Good morning, everyone. Respected dignitaries on the dais, faculty members, my dear students, ladies and gentlemen. Today's seminar, conference, or even workshop is based on artificial intelligence, and one more topic is co-crystallization studies. Actually, first speaker of this conference, Dr. Amit Pant, is from outside, and he is waiting in virtual lobby, and that's why actually we are 30 minutes late in this program, and he has to wind up this talk before 12 o'clock, if I am not wrong. So you all aware about the role of drug discovery or role of artificial intelligence in drug discovery. Yesterday, Honorable Prime Minister of India inaugurated one very important summit, that AI Impact Global Summit 2026 in Bharat Mandapam, New Delhi. From this summit is from 16th to 28th of this month, and majorly focused on artificial intelligence. And I have referred its entire brochure, and it is mainly on the well-being of human beings. Mostly many topic, because yesterday they deliver more than 28 plenary session throughout the day, and each topic is related to the main concept of that particular theme, and that is the very interesting one, that is 3 chakras, 3 sutras and 7 chakra. And I have prepared a short lecture for that, and definitely with the permission of Thakre sir, I will share that presentation share that lecture to you as well, because that is focused mainly on 3 sutras. First sutras is related to people sutra. People means एक आम नागरिक, how this artificial intelligence is useful for common person, start from agriculture, security, food, and even health. And that is very important, and even they have given some principles related to how we are going ahead with this sutra. And second sutra is planet sutra. That means environmental is also important, and with the environmental, if वो कहते हैं ना कि सपोज हमने एनवायरनमेंट पे बहुत ज्यादा यदि मैनिपुलेशंस किया तो एनवायरमेंटल crisis definitely will grow. Few days back, one conference is there in IPA College of Pharmacy on aerobiology, and in that role of aerobiology in artificial intelligence. So they have various models that gives how the pollen grains, how the antimicrobial resistance affect the social economic level of a human being. And that's why how this environmental aspect is very important as far as today's scenario is concerned. And third sutra is related to progress sutra. Progress sutra, even now एक या 2 साल में या 3 साल में we are the one of the largest growing economy of the world. And to achieve this, artificial intelligence is definitely one of the greatest pivotal role in the development of our economy. So these 3 sutras is definitely together shape the balanced philosophy that ethical, sustainable, and future-ready progress of the world with the help of artificial intelligence. And again, 7 sutras are there, and these 7 sutras is related to, first is human capital chakra, inclusiveness chakra, safe and trusted AI chakra. Next is science, which is very important. Then relations, innovation, and efficiency chakra, democratizing AI resources chakra, and AI for economic development and social governance and good governance chakra. And if you correlate, because when we are in the student life, or even in the beginning of our academic career, the drug discovery is only related to QSR studies, nothing more than that. Only we are going to derive QSR equation, but now various sophisticated tools are available, including a tool like SilicoXplore from SilicoScientia. And as Dr. Ataul may during the discussion may also convey, we are the one of the user of SilicoScientia at Department of Pharmaceutical Sciences, and our student regularly doing the various drug discovery or using various drug discovery tools for their regular research activities, and definitely it is very useful. And the next, their tool is co-crystal studies or crystal structure prediction properties, which is again the very important tool as far as SilicoXplore tool is concerned. Because why this tool is important? Earlier, if you look at our syllabus, हमारे सिलेबस में कहीं पर भी आर्टिफिशियल इंटेलिजेंस नाम का कोई भी चैप्टर, कोई भी ये है नहीं. ओनली इट इज लिमिटेड टू ड्रग डिस्कवरी. बट नाउ in one syllabus is there that is under the consideration from Pharmacy Council of India, and in that syllabus, because this is the proposed syllabus from Pharmacy Council of India, and in that syllabus means आपको ये लगेगा कि नहीं ये फार्मेसी में भी आएगा. There are some important subject like AI and Python programming for pharmacy student 1, again 2, AI in pre-formulation and formulation, AI in drug design, discovery and bioinformatics, AI in pharmacology and drug safety, AI in pharmacognosy and biotechnology, artificial intelligence, manufacturing, smart QA in pharmacy, and last is AI in pharmacy practice and patient care. This is the importance of artificial intelligence in pharmacy profession also. That means it is very essential to learn. Even at this age, we are also regularly updating ourselves to gain the various aspects of artificial intelligence in our research. I must congratulate Team SilicoScientia to develop the tools related to artificial intelligence and bioinformatics. Definitely, this one-day seminar and workshop and hands-on experimentation is definitely going to be a fruitful platform for all the young and budding pharmacists or young and budding researcher. Thank you, Dr. Vinod Thakre sir, and my mentor Dr. Pramod Khedekar sir, to giving this platform and opportunity to talk in front of you. Because of lack of time, I stop with the best compliment for the successful organization of this event. Thank you very much. Thank you.
Thank you, sir. AI is artificially intelligent and transforming pharmacy into predictive and personalized science. Sir has given this to us. Now I would like to call upon Dr. Pramod Khedekar sir to proceed for vote of thanks.
A very good morning to all. Now, first of all, I apologize that we have started half hour late, and now some jumbling is there, so we might shift that lecture from Germany first, and then we'll switch over to Amit lecture. Now, I am here before to give vote of thanks. Now, thank you is such a prayer that cannot be seen or touched, and it must be felt by heart. I feel honored and privileged to get the opportunity to propose vote of thanks and this special occasion of one-day international seminar and drug discovery training program. I would like to thank our beloved leader, Honorable Shri Dattaji Meghe, Chairman of our Sanstha, Shri Vidyarthi Sudhar Sangha, for their constant motivation and support to us. I also feel very much thankful to our dynamic personality, Honorable Shri Raviji Meghe, Secretary of our Sanstha. I would like to thank our young leader, Honorable Shri Samirji Meghe, sitting MLA from Hingna constituency. And I am also very much thankful to Honorable Madam Brundatai Meghe, Chief Advisor, Meghe Group of Institute, for our constant guidance. I would like to extend my sincere thanks to today's chief guest, Dr. Praful Sabre sir, who is my colleague, for sparing his valuable time and giving his guidance to our participants. I would like to extend my sincere thanks to today's guest of honor, Dr. Sushil Raut from Biocent Research Private Limited, coming from Pune and interacting with the participants. I would also like to extend my sincere thanks to today's online speakers, Dr. Amit Pant, research scientist from USA, Dr. Mahati Munich, bioinformatics scientist from Germany, then team from SilicoScientia Private Limited, Bengaluru, Dr. Ataul Islam, Director, then Vikram Singh Suryavanshi, Computational Chemist, Mr. Kunal Shire, Bioinformatics Scientist, Ms. Suhani Gavane, bioinformatics scientist. And here I would like to mention that all this is possible because of one of my senior student, Dr. Rupesh Chikle, who is research scientist, University College of London, School of Pharmacy, London, UK. Actually, he was supposed to come physically, but due to some administrative hurdle, it is not possible for him to attend this seminar. I would like to thank all principals of other colleges, staff, and participants who are here I am seeing in front of me. And I would like to thank our principal, Dr. V.M. Thakre sir, for his constant support at various stages for making this international seminar a grand success. I would like to thank all professors, staff members, supportive staff, all organizing committee members, especially Dr. Swati Gaikwad madam, Hina Mehta madam, Dr. Devare sir, and Kode sir for their hard work to execute this international seminar in Toto. I would like also thank our university, DBATU, IPA Nagpur Branch, APTI Maharashtra State, IQAC, then NCP at our sponsorer, Vico Laboratories MIDC Nagpur. From their support, we have got the kit. Last but not the least, I would like to thank all participants, those who have joined in offline as well as online mode. Now, the program, we have thought this last 2 months back, and it is very important that execution is possible only because of work. Without team, it is not possible. And I hope it will be a grand success. Thank you very much.
Thank you, sir. Now I call upon Dr. Swati Gaikwad madam to start the session.
Miss Mahati Munikoti, ma'am. Am I audible?
Yeah, I can hear you now.
Yes, starting with our invited speakers, invited speakers. Today, we gather on a common platform to exchange knowledge, share innovative ideas, and strengthen global academic collaboration in the field of research and development. So we are going to start with our first invited talk with Miss Mahati Munikoti, ma'am, Senior Bioinformatics Data Scientist, Mycoliver GmbH, Germany. For this session, she will speak on transforming pharmaceutical sciences through artificial intelligence. And I would like to invite Dr. Vaibhav Planchivar sir to session chair for this talk. Sir, please kindly introduce Ms. Smriti Munikoti with us.
Hello. Good morning, everyone. Welcome you all to the International Seminar on Drug Discovery. It is my pleasure to introduce Ms. Mahati Munikoti, Senior Bioinformatics Data Scientist based in Bonn, Germany. She brings over 7 years of experience in AI, machine learning, and multiomics analytics applied to drug discovery and precision medicine. Currently, she is working at Mycolever, Germany, where she is developing a fungal biocompounds discovery platform. She also has led AI-driven biomarker discovery projects in the USA and has published in reputed international journals. We are delighted to have her with us today. I now request Ms. Mahati Munikoti ma'am to kindly deliver her session and share her valuable insights with us. Please, ma'am.
Hello, everyone. Thank you, sir, for your kind introduction. I'm excited to be here. I hope I'm audible for everyone.
Hello, ma'am. Please.
Hello, ma'am. Hello, ma'am.
Yeah. I've shared my screen. Can you see my screen? So, yeah, today pharmaceutical development, from identifying new drug targets to predicting the crystal structures for formulations. I've designed this talk in such a way that even with somebody who has no prior AI background should be able to easily understand what AI can do in pharma, how it works, and why it matters. So today's roadmap would be like we'll start with the basics so that everyone is comfortable with AI terminologies. Then we'll walk through the entire drug discovery pipeline, and I'll show where AI can create impact at each stage. Then we look at how specifically at crystal structure predictions and co-crystallization, since that is the focus of today's seminar. And then we'll also discuss about the challenges and ethics. And also feel free to interrupt me in between if you have any questions. So this slide just paints a picture of why AI matters in pharma. So these three numbers can pretty much sum up the entire story. So first, to bring a drug, the average cost is about, it costs about $2.6 billion, and it takes about 10 to 15 years. And also about 90% of the drugs that enter clinical trials fail. That is about 9 out of 10 drugs that passes all the preclinical steps but still doesn't make it to the market. And these numbers pretty much tell how unsustainable this is. And think of it like studying. The more examples you see, the better you understand. That's how machine learning is. And the innermost doll would be deep learning. So deep learning is like the innermost subset of AI. So this deep learning is inspired by human brain. There are many neural networks and with many layers. This is used for understanding complex patterns. So where it works on every step or achievement, we give a reward. So every good outcome gets rewarded, and based on these rewards, what AI does is like it tries to perform different actions, and the ones that are like the correct, it gets rewarded. And this is used for designed for optimally for like designing new molecules. That is where this helps us, reinforcement learning. And then generative AI, this is something we're all talking today. This is the technology behind tools like ChatGPT. It can create entirely new things. In pharma perspective, we can say it can help us in designing a novel drug molecule with our desired properties. Then we'll see why pharma needs AI. As I said previously, it can help us reduce the time, cost, and accelerate. Like how? So there are four, I would break this down into four critical reasons. One is we're drowning in data. So a single genomic study can generate terabytes of data. So we need some powerful machine that can analyze, because definitely humans can't analyze so much of data. And then we have clinical trials, electronic health records, published literature. We basically have so much of data. So AI can help us analyze all of this at a very faster pace. Then speed. So COVID-19 was like a wake-up call for all of us. We were able to develop vaccines under a year, but that was just an exception, right? So this should become a routine, and AI can help in making this speed a routine for all of the drug development. And then third would be complexity. Human biology is very complex, and protein fold in billion possible ways. There are so many drug interactions cascade through interconnected pathways. So traditional approaches can't capture all of this complexity. And then fourth would be the chemical space. So there's n number of possibilities to estimate drug like molecules. And even if we like search say one molecule per second, it would take so much of time, larger than the age of this universe, to check all of them. But AI can intelligently navigate this space and find the promising candidates. Then, if we look at how the traditional drug discovery pipeline is, it's very long, time consuming. So it starts with target discovery, that is identifying which protein or pathway causes the disease. Then we that would take about 2 to 3 years, and then we go to the lead identification, finding the molecules that interact with this target, and then optimize it. Make sure we have to, like, improve or tweak the molecule to improve the properties. Then we do the preclinical testing that happens in 3 phases, which alone takes about 6 to 7 years, and then finally the FDA review. So this is like, the funnel is incredibly narrow, and for every 10,000 compounds that look promising in lab, only one makes it to the pharmacy. So that is about 99.99% of failure rate. Now we can imagine how AI could make each stage faster and more accurate, and that's exactly what is already happening at companies. And here, the same timeline, we'll see how AI can transform it. So at every step, so let's say target identification, it analyzes the genomics data and the protein interaction networks, and it finds about the new targets 50% faster. And then identifying the hits, generative AI can even screen about 100x more compounds virtually than traditional high throughput screening. And during the lead optimization stage, AI can predict the ADME properties, and it can also reduce the computational. It can predict the ADME properties computationally and reduce the cost about 60%. And in preclinical trials, AI models can, predict toxicity, adverse effects before even going for expensive animal studies. And when it comes to clinical trials, we can utilize AI to stratify the patients to find the right population. That can also help the study researchers boost their success rate by 30%. And for the regulatory submissions, we have natural language processing models that can read through the documentation and also help researchers navigate through the entire process. One key thing would be, we're not saying AI is going to replace pharmaceutical scientists, but it should supercharge them. You still need the domain expertise, but AI will be used as a tool to that can, like, boost the scientists. Then let's see, like, when we talk about in detail, like how AI can help in target identification. So the first step in drug discovery is always finding the right target, and usually a protein involved in causing or sustaining a disease. So AI can revolutionize this in 4 ways. One would be network pharmacology. Uh, AI uses We can use AI here to map the entire web of protein interactions and analyze this in silico, and then identify the hub proteins that play very important role in disease targets. So this is this can be missed when we do single gene studies. We might miss this, but AI can fasten this, and we can understand how these interactions are. And then it can also help in integrating the multi-omics data. It can simultaneously look into genomics, transcriptomics, proteomics, and metabolomics data, and find targets that are supported by multiple lines of evidence. This multi-dimensional view actually increases the confidence in target selection. And as I already said, we can utilize NLP to, like, do literature mining, scan through millions of published papers, and then AI can extract this information. It can give you a summarized knowledge of what is needed, and then we can build these knowledge graphs that can connect all of these biological knowledge. Like, you can have a queryable network, and then this can be later used to validate in clinical trials as well. And then how AI can help in drug design and molecular generation. So there are two main approaches. One would be the de novo drug design, where AI can help us create entirely new molecules from scratch. Uh, so here we have certain models called variational autoencoders. These right now everyone's moving towards generative AI, but these GANs and VAEs, these are the models that started with this generative AI concepts. They started with these primitive models. These are now, we can call outdated, but people still do use them. They're powerful, but now everyone's using transformer based models like ChatGPT or generative models, ChatGPT, and all of these for developing this de novo drug design using AI. And the other part would be, we can look at virtual screening. As I said, they have a huge, like, it can rapidly evaluate the existing compound libraries and then screen at a 10x rate, and we can get the results at a very faster pace. Then for ADME, traditionally we synthesize the compound, then spend months on testing its ADME properties in labs. But we already have certain models that can predict the absorption, the bioavailability, the distribution, whether how the drug spreads through the body, the protein binding, blood-brain barrier penetration, and all of these can be predicted. And we already have such models or tools like ADME Lab, uh, Swiss ADME, PKCSM. They all use AI to predict these ADME properties. And then in clinical trials, it can help again in four different ways. One would be like about 80% of the clinical trials fail. Also, they face this delays in recruitment. So AI can help in analyzing these electronic health records across the hospital systems, identify patients who meet the eligibility criteria, and then this can help the recruiters like a time of about 30 to 50% of their time, instead of looking for the patient cohorts who they could test their drug on. And then there is companies like DeepSeek's AI and Tempest. They already do this, and it can also optimize the clinical trial design protocols. Instead of like having fixed protocols, it can be very adaptive. It can adjust based on the interim data and everything. So it can be very flexible. And then it can also people are already using AI or ML to, use this for biomarker discovery, and this also enables precision medicine. And finally, it can also help in monitoring the real time data from wearables or patient reports, or if it can detect any adverse events earlier. Yeah. And then when it comes to formulation, what AI can do is, on the left, we see that it can help in predict which excipients work the best together. And it can also, like, the models also can tell like how the drugs will be once it is like released. It can help in optimizing the manufacturing process, predict the stability under different conditions. We can also design experiments efficiently using design of experiments, and it can reduce the failed batches by 40 to 60%. And in real world, on the real world applications for like solid dispersions, AI selects optimal polymer drug combinations for amorphous solutions to improve solubility for poorly soluble products, drugs. And for nanoparticles, ML models can optimize multiple parameters simultaneously. We have 3D printed tablets that represents, this would be like the future of personalized medicines, where people are working with 3D printed tablets. And then for crystal structure prediction, there are different how do I put this, like for the same drug we can for the same drug molecule it can crystallize into different arrangements, right, different polymorphs. So these different crystal forms can have dramatically different properties. The dissolution rate can vary by 100-fold uh, between 100-fold, and they directly affect the bioavailability. So I think one of the most famous example would be ritonavir. In I think 1998, Abbott's HIV drug suddenly started crystallizing in a new polymorph that was less soluble and less bioavailable, and they had to pull the product the market, and it cost them about $250 million. So the patent protection also depends on the crystal form. How does AI predict crystal structures? Traditional CSP is computationally very expensive, and it requires exploring millions of possible molecular arrangements. So here, ML, we can use ML models that has these force fields and appropriate quantum mechanic calculations to like fraction the cost. And then we have graph neural networks that can identify how these interactions or how the molecules interact with each crystals. And so there are a lot of different models that can help us study this or predict this stable polymorphs for a drug before we even make a single crystal in the lab. And co-crystallization studies, it's also like a very powerful formulation strategy, right? So it can AI is making it more practical. And in pharmaceutical, co-crystal contains the drug molecules and the conformer molecule. They're held together by the hydrogen bonds. But the beauty of co-crystal is they can dramatically improve solubility, bioavailability and stability without changing the drug's structure. Right? Traditionally, finding the right conformer was a trial and error. Scientists would screen.
Ma'am, please on your mic. Ma'am, please on your mic. You are not audible.
Ma'am, please unmute yourself.
Am I audible now?
Yes, ma'am. Yes, ma'am.
Yes.
Okay. So, as I was saying, for co-crystallization studies, we can use a lot of these. There are already a lot of models that have been developed, and some of them use this Cambridge structural database, and it can predict conformers that most likely to form stable co-crystals with your drug. And hydrogen bond analysis also uses AI to like analyze the molecular electrostatic potential surfaces, The red and blue maps you see in chemistry, they're used to predict where the hydrogen bonds will form, uh, and which synthons are more favorable. And then the case study I would like to talk about AlphaFold, because this is a very famous example, and last year we even won a Nobel Prize for this, right? So for 50 years, the protein folding problem, like predicting a protein's 3D structure from its amino acid sequence, was considered like one of the biggest challenges in biology. And in 2020, DeepMind's AlphaFold 2 essentially solved it. It predicted protein structures with accuracy. rev- Uh, so it was, I think, extensively like how we have the X-ray crystallography and cryo-EM. It stood there, right? And AlphaFold predicted structures for over 200 million proteins, virtually for every known protein. So this the accuracy is sub angstrom for many of these proteins. So this is a very I would say an achieving feat, and this all started with the back end. How the AlphaFold worked was the basis of AI, the different deep neural networks, the transformers. Now we have predicted nearly, like, I think previously there were only about 17% of human proteins that had like experimentally determined structures, but now with help of AlphaFold we nearly have structures for all of the proteins. So this opens up entirely new drug targets that were previously impossible to work with. And then AI driven drug discovery platforms. And today you will be learning about SilicoScientia, SilicoXplore, how this platform works. Apart from this, like, we have atom wise, Schrodinger, the recursion pharma, they all use all of these platforms use different AI modules and models, and each of them have their own use cases as well. Schrodinger combines both physics based simulations with machine learning, and recursion pharma, it uses like a computer vision approach, because they analyze millions of microscopic images of cells to identify the drug effect at a specific level. And then AI can also help in drug repurposing. This again, when we want to study these compounds at a very when we study these compounds at the interaction level, AI can even accelerate how these drugs might interact with other targets, and it can be used for repurposing. And then personalized medicine, this is the future, like this is where we are headed at. And each for like the pharmacogenomics, the companion diagnostics, dose optimization, and then finally everyone's even already talking about digital twins, but this is where we are headed at. And eventually each of the patients or the cohorts will have like individual digital twins that can help map. And then this is where personalized and precision medicine is going to boom. And that's this is the path that AI is going to help us reach. And then we can even have its applications in quality control and manufacturing. For all of these processes we can use AI to automate. And as I already said, we can make use of natural language processing. for to go over the literature, understand the paper. It can even like read, understand, extract structured knowledge from all of this literature and help us finding drug disease relationships, the mechanism of action, potential biomarkers that could that would take human researchers. Then, when we talk about computer vision, we have like a lot of these image based, right? Like histopathology, the cell images, the tumor images. For all of these, like the medical imaging, for all of this, we already have certain vision based models. And when these assays or analysis generate so many images, these AI can like help us process like speed in the process of identifying which or detecting the nodules through the scans, or detecting minor invisible, I would say, smaller tumor or any kind of issue that with eyes we can't just look. The AI can help
रील भी
understand.
नहीं आया था ना था हाँ ना हाँ माँ
And even in regulatory affairs, as I already mentioned, we can like it can help us understand the technical documentation, how to standardize these and maintain the compliances. All of these can be
माँ चल रहा है
text and monitored using AI. And when it comes to like the market size, AI in pharma is projected to exceed about $50 billion by 2028. It is one of the fastest growing segments in healthcare and technology. And there are about 150, I think about 150 AI discovered drugs currently in clinical pipelines across various therapeutics. And companies using AI report their hit to lead timelines are about 4x faster than the traditional approaches. And AI has optimized the clinical trial designs about 30%. And several top 20 pharmaceutical companies are already using AI in their R&D. So it is no longer experimental, it's mainstream.
Ma'am, please on your mic. Other all participants, I request to close your mic. Please switch off all the participants your mic. Thank you.
Uh, can you hear me now?
Yes, ma'am.
Yeah. So as I was saying, these are the tools that anyone, any pharma students or anyone who wants to like just explore and understand how AI can help in their research. These are some common tools, AI tools that can be used. And then, natural products are like the treasure trove for farmers, right? 60% of approved drugs are either natural products or derived from them. So even here, like with when it comes to genome mining, the spectral matching, retrosynthesis, the bioactive predictions, and understanding the metabolics. Right now, everyone's building these in silico or computational metabolic models as well to understand how these mm, the pathways, like how these compounds interact and what pathways are triggered, to understand all of these things. And then for when it comes to generative AI and large language models, these are like the ChatGPT like models. These for like generating new molecules or creating hypotheses on these generations as well, and also the literature synthesis. then we spoke so much, but the main there are some challenges or limitations, and the main challenge I would say is data quality. Uh, so pharmaceutical data or the data that these companies generate, the researchers generate, they're often incomplete and biased towards a certain medical scaffold, and they're noisy, and so much of it is a proprietary data. And then there is also this black box problem, right, for regulatory acceptance. But right now we do have certain explainable AI models, and people are like warming up or trying to accept these models. But still, there is a huge gap where we don't entirely trust these models because they don't know why or how why these predictions are made. And then there is a gap in the validation. There is also like a problem with reproducibility, right? There are a lack of standard benchmarks or reportings, and to compare different AI methods and reproduce. So this is another uh, gap. And then also uncertainty with the regulatory, right? We don't know the frameworks that FDA, EMA, they're all working at, and nothing is finalized. So there's always this uncertain. And also there's a lack of there's a shortage in talent, because and also I think this is where the biggest opportunity lies, because we would need a we would need talent who has expertise in pharmaceutical sciences but also understands AI. And an amalgamation of this is very rare, and it's also very valuable. And that is how everybody should like think, because whatever expertise we have, we need to have like certain knowledge of AI, and that will just uh, makes us unique. And that is the kind of talent that the industry is also looking at. And also, we need to consider the ethics, like the patient data privacy, GDPR, bias and fairness. All of these things are very critical, and we need to have these in consideration whenever we are working with any kind of AI model. And then what I think is coming in future is like now we have these AI assisted tools, and then later in we'll have an end-to-end AI platforms that we're developing right now that can manage the entire drug discovery campaigns with minimal human intervention. Then we have autonomous labs already. There are people are working on these robots that can execute different experiments and everything. So we will in future have these self-driving labs. And then comes the precision everything. As I said, like individual patients will have their own digital twins, AI designed personalized medicines, real time health monitoring. So the key takeaways from this talk is AI is transforming every stage of drug discovery. The crystal structure prediction and co-crystallizations are being revolutionized by different ML models. And AI is not replacing scientists, it just amplifies the capabilities. And always we have to keep in mind the ethical consideration, and the main challenge would be the data quality. And also the industry needs professional who can bridge science and AI. Thank you.
Thank you, ma'am. For any queries, please raise your hand. Offline mode, online mode. Mr. Bhushan Pahune, ऑनलाइन मोड से भूषण पाहुने उन्होंने हैंड रेज किया है. हेलो हेलो मिस्टर भूषण पाहुने, you have any query? My mistake.
offline mode. Anybody is having any query?
Hello, am I audible?
Yeah.
So actually, whatever presentation you are giving is really very nice. My question is that as you told that there are a lot of AI-based thing is come and scientist is not replaceable by the help of AI. So my question is that is there any drug which is on the clinical trial which is AI-based?
Yes, there are some drugs, like about, I think, 18 drugs. I don't remember the name, maybe about 18 drugs that are in clinical and preclinical trials that are
Not
discovered.
on which clinical trial? Clinical first, second, or third, or is it marketed?
No, no, it's in the later stage. So it is in second and third phase.
Okay, so that drug is used in which disease, like particularly on the TB or any other?
Mm, Let me look it up because I have it here. I think it's by In Silico Medicine. If you look at In Silico Medicine, they have a drug for I'm not sure which disease, but they've published one of them is in the clinical trials.
Yeah, in silico based drug is on the
And
clinical trial or preclinical trial, but in the AI based it's not possible because in the AI based they only give some data that if the data you do the in vitro or in vivo analysis, uh, the data. I also tried one of the drugs, C5 I also use. Okay,
Mhm.
so that is failure on the preclinical trial. So I'm not sure. I'm sure that in the AI-based drug, I think is not available right now.
Yeah,
But
actually,
that's
Mahati, just a minute. Yeah, this is a good question, whether AI can develop the drug or not. So AI is fully can't develop the drug, but it can help to accelerate the drug discovery pipeline. And maybe in the before lunch, I'll show you around 30 to 35 molecules are in clinical trials. Those are developed using the AI and machine learning. So if you are saying that is yes, without any experimental, it is not possible, only the computational to discover drug. And yes, it is also true that is the no drug approved yet through the AI. But all of the lot of drugs, lot of molecules at the latest stage, as Mahati said, are in clinical trial. Those are came from using the AI and machine learning approaches. So maybe I'll show you more data before lunch.
Okay, sir. Okay, sir. Actually, in the machine learning, first you have to train your data. So if you are having a lot of data, you train the machine, and after that, it gives it follows some algorithm. So on the basis of follow the algorithm and scoring that, then they sort out that this drug may be the alternative for this disease.
Yeah, definitely. But ultimately, you have to go to experiment. So if I saying, if you're asking me that is whether I can develop a complete drug to the AI based, it is not possible at all. So AI or machine learning is a mediator which can help you to accelerate the drug discovery process. It can reduce the years of time and huge amount of the investment. That is the beauty of this approach. I hope you.
Yeah, you are right. Yeah, you are right. Correct. I agree.
One more question.
Mhm.
Good afternoon, ma'am. My name is Ravin Singh Rathore from Nagpur College of Pharmacy. My question is that, ma'am, how AI helps in clinical trial like biomarker discovery? What can you explain what is biomarker discovery?
Mm-hmm. Sure. So when we talk about biomarker discovery, right, like we do a lot of these gene expression analysis and identify certain genes or markers in studies, right? To do all of these analysis and identify, pinpoint these genes, we do use a lot of these machine learning algorithms, because when it comes to studying about this expression data and everything, the data is very heterogeneous, and that's where like AI can help us uh, modulate this data and then understand. So that is like would be the starting point of understanding the genes and the markers for a particular study. And also there are a lot of these AI models that are being trained on these single cell data, very particularly, like say if you focused on pancreas, then you could study that the model is already, we have models that are already trained on this data. So you just need to like look that particular model, give your data what you have, the your area of interest, and then identify the key genes that are involved in this. So that will speed in the biomarkers discovery. And also there are automated pipelines and tools that can help you do the gene enrichment analysis and the pathways, the mechanism and everything. So there are already automated ways, and this can just help us at least pinpoint the key genes or key markers for a particular disease. That is how AI is helping us in the biomarker discovery. And when it comes to clinical trials, there are certain modules based on the clinical trial design, the study, the inclusion exclusion criteria, and all of this. There are models that can already help researchers whether their clinical trials is going to succeed or not. If it's not succeeding, what are the causes? How they can, you know, amend their design of the study design and all of this. There we already have certain models.
Okay, thank you, ma'am.
Thank you, ma'am.
Uh,
Due to the lack
yes,
of
ma'am.
time, I
Can you
request
hear me?
all of you to send their questions on
Sir, I asked last question.
Yes, sir.
Hello. Yeah, ma'am, you told me right now. You told that regarding clinical data and AI,
Mhm.
there is a question that many time it was seen that generally person train its AI on the like US origin patients or European origin. Okay, it will not same if shows effect in Asian origin or Indian origin. Then
Mhm.
how we can minimize that?
So what we can do is, and this is something that like how the research happens in like say India as well. We have a lot of data, but we don't have the studies that are like for specific for Indian population, right? So what we do is this: there is something called as foundational models. So they are like broadly trained on all the information that's available, like say US patient data. So we use these as a foundation, and then we have like our Indian patient data. We fine tune this model specifically for that. So what this does is, the AI already has some information or knowledge on how this data is, right? And for that we feed in our like the regional information. Like say for Indian population we have about 100 or 200 patients information. So we train the model and ask it to like specialize on this, but it already has the prior knowledge. So that is how we're working with other demographics.
Okay, thank you.
Yeah.
Thank you, ma'am. So we are concluding. As we conclude the insightful lecture, I sincerely thanks our distinguished speaker, Ms. Mahati Munikoti ma'am, for enlightening and thought-provoking preparations. I appreciate the active participation of our attendees and dedicated efforts of our organizing committee in making this session successful. Let us apply the knowledge gained today in our academic and professional career. With this, I officially declare the session is closed for Mahati Munikoti ma'am. Thank you, ma'am.
Thank you. Thank you.
With this wonderful session, we are moving towards our keynote session. So, every successful gathering is guided by visionary thoughts, and the keynote address is a soul of that event. The keynote session sets the direction, inspires curiosity, and opens new horizons for meaningful discussion and scientific advancement. So let us start with our keynote talk by Dr. Amit Pansar, who is a research scientist at Creighton University, USA. And to chair this session, I would like to invite Dr. Praveen Kumar Surse sir. Now, may I request Surse sir to introduce our keynote speaker, Dr. Amit Pant sir.
Good afternoon to all. Respected dignitaries, distinguished delegates, and dear participants. It's my privilege to introduce our esteemed speaker, Dr. Amit Pant, currently serving as postdoctoral research fellow at Kington University. Dr. Amit Pant is a distinguished medicinal chemist with strong expertise in bioactive molecule synthesis and drug discovery. He has a proven track record in hit-to-lead optimization and drug candidate selection. His research strength include rational drug design, computer-aided drug design, structure-activity relationship studies, and molecular modeling techniques such as docking, 3D QSR, and molecular dynamic simulations. He is also highly proficient in modern analytical techniques including NMR, LCMS, HPLC, GC, and IR spectroscopy, along with advanced biological assays and in vivo models. He has completed his PhD in Pharmaceutical Sciences from RTM Nagpur University and his M.Pharm in Pharmacology from Dr. MGR Medical University, Chennai, and B.Pharm from Sant Gadge Baba Amravati University. Dr. Pant has held prestigious research position including a CHS postdoctoral fellowship at University of KwaZulu Natal. His excellence has been recognized through several awards including an industry-sponsored research grant, the CHS postdoctoral fellowship, and the international travel grant from the Volkswagen Foundation to attend the Nature Conference on HIV immunity and eradication in Hanover, Germany. He holds 5 patents including recent United States patent application 2024 related to novel trifunctional AMP analogs with ocular hypotensive and neuroprotective effects. His research contributions include 15 publications in SCI Index Journal and 4 book chapters. He has also presented his work at prestigious international platforms, including the American Chemical Society Midwest Regional Meeting in the USA and the conferences in Germany, Chennai, and IISc Bangalore. With his extensive experience in medicinal chemistry and translational drug discovery, we are indeed fortunate to have him with us today on online mode. May I now request Dr. Amit Pant to kindly deliver his talk. Over to you, Mr. Amit sir.
Thank you. I'm confused now what to say. It's good afternoon, possibly, because here is a midnight, almost 1 o'clock right now. Am I audible to everyone? I
Yes, sir.
hope so. Thank you. Okay, so I was supposed to start, I guess, 11 p.m. of the US time, but it's got delayed. But anyway, I just want to again, to not take much of the time, and I just want to jump on the discussion. I'm trying to talk about AI-driven drug discovery, the reality beyond hype, because we are a lot of talk everywhere. We are getting into the discussion of AI, but what actually the reality, and is it really benefiting us? So before we start, let me go ahead and introduce you something, from where this is my audience sitting in the NCP and around the other corner of the globe. I'm talking from you in the Omaha. This is the actual campus, and you can see on the corner building here, I am sitting in this building and I'm talking right to with you. The Creighton University, I just want to briefly introduce you. This picture is everyone well aware. The Kalpana Chawla, they lost their lives in the mission on in the space. So one of the Michael P. Anderson, the astronaut, he was the Creighton alumni, and we are from the Omaha. The well-known personality from Omaha is Warren Buffett. He is the among, I think, top 10 still in the richest person list in the world. Uh, this is just a brief background. So what we're gonna cover today? We're gonna have very quick review for the AI. We will talk more than about conventional drug discovery process. All the pharmacists are well aware about this, but still I'm gonna touch in respect to the AI as well, how that AI is transforming drug discovery, and specifically for my topic of discussion, I will focus more on the target identification and then actually drug discovery using AI. And at the end of presentation, I'm gonna give you some of the useful resources. Few are freely available for you because I was just reading some of the comment from the previous talk. The You are already interested what resources you are available. One is already there in between you, the industry resource who is gonna talking about it. But okay, so what is AI and artificial intelligence? Always when you see AI, any icon, any logo, you will see one like it is correlated with brain and as well as intelligence word itself indicating something. So let's have a brief introduction for the AI. It is a definitely branch of computer science, and that's why AI gimmick is giving them a more pressure than other pharma industry personnel, I guess. And intelligence wise, that's why, because AI is also trying to adapt themselves in a learning capability, reasoning, problem solving, perception, as well as decision making. The AI has a very strong ability for vast data analysis. It can identify patterns and improve performance. I want to highlight the type of AI. There are three type of AI. One is a narrow, which is what we are currently experiencing, because they are task specific, field specific, or they are designed for a specific job to finish. General AI is something which we have not achieved. So general AI typically is a system who can do everything. So it is not only the task specific, but it can accomplish multiple tasks at a time. And super intelligence, I think we are right now out of that's only the theoretical part right now. So I think I just got a message that can I share myself? Can you visualize me? Excuse me. Can you see me?
No, sir.
Can you see me right now?
Yes, sir.
Okay, good. Okay, so ultimately, AI is nothing but the mathematics and the neuroscience. So because the programming is doing uh, based on the neurological science and intelligence, that the term. So okay, so moving ahead, let's see. Oh, it's AI. Oh, no. It is like AI and you. Yeah, that's the right. So what is the AI and you? So this is all around surrounded with all the gadgets. They are controlled by and supported by AI. Simply like Alexa, your Gemini from the Google, Siri from the iPhone, Map, Netflix, everywhere. And the advanced version is the automobile, like you can self-driving cars. That's the recent most advanced version of the AI, which is around you. I think we are missing best important part is a brief history of the artificial intelligence. So I'm just gonna show you some of the major key highlights in it. artificial intelligence. So in 1950, that's the era where we got independence. So Alan Turing, who just proposed a thought, can machine think? And the hypothesis he wrote a published article into the journal called Mind. So that is the very first conception of the artificial intelligence, though actually this term was coined into the Dartmouth conference in 1956. And after that, okay, I guess there is a sound issue. Anyway, so in we are almost past 75 years and we are into the advanced era of AI. So the biggest achievement was from IBM's Deep Blue model, who developed a chess playing kind of program, and that program beat the chess champion Gary Kasparov. That was in 1997. So just within the first 50 year of the AI era, and now we are in the very advanced era where most of the things are capable by the AI. So how it works? We have seen many of the terminology, algorithms and all, but actually AI process a data which is available in the public domain. Some paid AI softwares or tool they used, maybe paid journals, paid articles, patent, which are not publicly accessible. So they have the deep uh, data analysis. They also have the ability to simulate with the human cognition. Like if you see autopilot car, they can drive you as like humans can drive. So what actually key technologies behind AI are? I think Mahati madam already discussed on these machine learning, deep learning, natural language processing, and I am not gonna take much of the time out of it. This is a timeline every pharmacist, I think, is well aware about the drug discovery pipeline. Target identification, then drug discovery, then comes preclinical, all the promising candidate till this stage, then they forward towards the clinical trial, and once they are satisfying all the criteria, approval is given, and then the last phase is a post marketing surveillance. So drug discovery is nothing but finding a needle in a haystack. It is a very complicated task because disease itself are multifactorial, and you know of- often identifying a treatment is key challenge. We have complex drug development process because we have seen there are multiple stages, and setback can come at any stage. So this is like a snake and ladder game. You can try to get to your target, but anytime you can come down at any stage. So in a drug discovery, if you fail into the clinical trial, you have to again come down to find out best hit compound because of the pharmacokinetic or dynamic model. If it is a reason, if a target itself is a problem, then you have to start from the beginning. And this is the chemical space which is available. So in a drug discovery, you have to explore the number of compounds. Just imagine, and I would like to show you that number of chemical compounds are expected to exist is almost in between 10 raised to 60 or 10 raised to 100. So it's not an easy job. Once we do all this, there is regulatory requirement which are very stringent, and they are often time consuming plus costly. So AI driven drug discovery, what actually it is, and how they are uh, helping in this pipeline, how it's transforming actually. So revolutionizing traditional drug discovery methodologies, and it is providing a very efficient option, because if you see, these are the multiple options and multiple stages where AI can optimize each and every step. And the most important is they can enhance accuracy as well as uh, whatever the prediction you are drawing with the help of AI, they are more accurate with the time saving option as well. And I'm just trying to figure out some of the numbers here. If you see, uh, the budget till only the preclinical research, the conventional drug discovery usually requires almost around 600 to 700 million US dollar. With AI, you can bring that budget to just 2 to 3 million maybe USD, and time is most significant because 8 to 10 years with your conventional method, and with the AI it goes to almost like 18 to 24 months. So considering setbacks probability in the research pipeline, these factor is most important because then you are still in a race. Now we just driving deep into the drug discovery using AI. And as I mentioned, I am first taking care with the target identification. So why it is most important and target identification is how crucial. So whenever you try to identify any target, it has to be a closely related and strongly related with the disease what is of your interest. It has some established role in its pathology, because if you are trying to find out some inhibitor agonist, so it has to do something with the disease. Most important, whatever protein or enzyme you want to target is actually it should be druggable, like some inhibitor agonist or whatever the chemical entity, they should bind it, and then they can produce the desired activity. And whatever you are inhibiting or activating, there should be some assay you have to either design or they should be available. There are conventionally multiple options available for the target identification, but most commonly pull down or the whole genome knockdown process are commonly used. It tend to be very time consuming, labor intensive, and high failure rates because it is a blind game. You don't know anything and you are just searching. But let's see. Okay. Just as in a brief, I think previous session also mentioned about the number of human proteome. So three-dimensional structures, almost more than 100K are available right now. Disease-related proteins, structural elucidation out of these 100K is close to 4,500 proteins. And out of these proteins, only 854 proteins, they are actually targeted for certain diseases. Whatever the FDA approved compounds are in a market, they are only targeting these 854 protein out of whatever the disease related proteins are. So you can see what and how big the gap is, what is the ratio of unexplored protein structural diversity available, and that's how your scope is. Because all the proteins could not be target. There are reason because they are difficult to drug targets. They don't have properly all the characteristic of binding pocket or the active site. They function via protein-protein interaction. So certain times targeting or blocking cannot produce any mm, desired activity. And these maybe certain are not even tapped right now. So I am trying to show you here in a figure the general comparison by three different approaches. One is the experimental, as I mentioned, pull down assay or knockdown. So that is completely actually we have to do experiment here. The other one is omics identification. So when I say multi-omics or only omics, it includes all the 5 branches of the omics: genomics, epigenomics, transcriptomics, metabolomics, proteomics, and all. And there is one computational related where actually your AI is playing role. But still, I am just showing some of the examples here which are generated with each and every method. Each and every method still holds some strong value, and they require certain expertise as well. Because if experimental process do not generate any data, AI cannot analyze and they cannot predict much more. So there need to be have some of the data and data bank as well. So experimental are still very important. So just for an example, I am giving you a brief representation through the various literature and data mining. If AI giving you a target like E3 ligase, so through AI you can see again, okay, E3 ligase is my target, how it binds with other protein components. So AI can then analyze, okay, there are multiple binding partners for the E3 ligase, and actually AI itself can again go dive deep and see these interact with certain which of the proteins, and then these width itself you can see like bromodomain containing protein. So they have been explored, and I think more than 100 they are been uh, analyzed till date, and that's the reason now AI has analyzed this data. You can retrospectively see it is predicting, and that's actually resembling with the research. Talking one more important thing for the protein here. All you know, proteins are amino acid chains. They have specific sequence, and maybe they are certain conserved within the family or pathogens. So, but just for an example here, this is a structure of protein, and this is its sequence. So highlighted yellow, these are the amino acids which are very important for protein function. Now through the AI, what this information is used, if this sequence is getting aligned into specific orientation and this sequence is accomplishing certain function. So if this sequence is available into other protein, do they also have the same function? From the data, AI then analyze a very vast data from the genomics, proteomics, and it is very easy to classify protein or select the protein from another family as well. I am now giving you some of actually data from the published articles, which you can correlate how the AI has been actually used, and what are possibilities of the AI in the drug discovery. So before this is just an article, and I think some of you who are interested, you can then refer down those articles as well. So this article is specifically talking about the AI in the drug discovery pipeline. We already discussed this, but actually I just want to highlight this specific portion here. If you consider the cost in the very early stage for the target identification, it is maybe up to 100 million cost is required, but AI can take it down to zero point 0.6 million. Significant reduction in the target to hit and hit to lead, as well as lead optimization throughout this process. You can consider here whatever the significant reduction of the cost using AI. And that's why for the pharma at least, application of AI is always beneficial. And I'm not going to talk about the other prospect right now, but here is what is important for our target identification discussion is AI integrates vast multi-omics data or whatever available in the literature, because if any master researcher is trying to accomplish a specific part of project, how many literature, or if someone in a PhD program, if retrospectively he want to analyze how many literature he have cited in his master thesis, there are maybe some hundred of literature. So you can see one person can only have accessibility to certain limit. They can read, they can understand limited literature, but this AI, they can screen a very vast data. Now the other important thing is, when you talk about omics, when I talk about the proteomics language, it's very difficult for understand by maybe a analytical person. Genomics language, if I am talking, it is very hard to understand by a microbiologist, possibly. So it is obviously our limitation, because we are focused with one as like bachelors, they are studying the basic of the sub field. Master, you are climbing one step up, and then in a research actually you are very narrowed down to specific topic. But now using AI, you can screen all whatever you required. If you required proteomics data to be screened, you can, with the AI module, you can select proteomics as an input. AI will do it for you. So that's how the impact of AI for us. And once we process the AI, process the data, we have option, like I'm talking about specifically for a target, in connection to the target. So If I want any, for any filter, I want to filter my target based on novelty. For an example, I want to identify a novel target from this data. AI will process, it will prioritize, and then it will give me the output based on my applied filter. So for me as an individual, I do not need to screen, read, and understand each and every article available in a database to understand the novelty. I can set it the parameterize into the AI. AI will do it for me. So it is giving me a much of ease in my research. So they are important, and they are now transforming target identification. I hope everyone is understanding how these actually correlating with you. I'm trying my best now. So this is another research article, which is a small molecule TNIK inhibitor target fibrosis in preclinical and clinical models. So they you in this article they reported from the target discovery, like how they use these different AI modules and they selected the novel target, how selectively they uh, design a compound who can target these specifically identified target, and then they have established its role in the disease pathology as well. So AI giving you very precise and accurate data from the target identification to your drug discovery step into the, so the probability of getting success into preclinical and the clinical trials is increasing by multiple folds. So these are multiple methods you can refer into the article how they used. So they uh, applied the novelty for their filter, and I am trying to highlight here is whatever for the disease reported, they are mostly tyrosine kinase family. But with these filters and AI, they also predicted one of the TNIK enzyme which belongs to serine threonine kinase family. So this is a novel one. No drug has been yet targeted this one. Like nintedanib is a one of the drug, but it is targeting this family, tyrosine kinase family. But this is something novel target they have identified, and they design a successful drug which is inhibiting this target as well. There is another example, AI-driven drug target screening platform identified oncogene activated by enhancer infestation in Epstein-Barr virus associated nasopharyngeal carcinoma. So here, this flow diagram, I just want to show you how this AI, so they use Pandaomics, the AI module, into their research, and they shown their success or the target validation into in vitro and in vivo model as well. So it is not only AI is giving you data and you know it's just like theoretical. Based on AI, once they validated target into the actual experiments, they found the accuracy and higher predictability. Now we are moving into the next step called AI in drug discovery. So we have covered the target identification right now. Let's move towards AI in drug discovery and what stages, what are different factors AI can impart in your discovery, and how you can boost these drug discovery, specifically hit to lead, then lead optimization, and preclinical research as well. So drug design, lead optimization, it can give you the entirely new molecule like de novo design rather than just searching the existing library. Virtual screening, so you can have a lot of number of compounds. There are multiple database, the NCBI, there is a database called zinc, so they have multi-million compounds into it. So experimentally it is beyond possible, but artificial intelligence can give you the option. You can screen them, and whatever based on the prediction, then you can think using your medicinal chemistry knowledge, and then you can shortlist some of the analogs. You can try to synthesize them, so you are curtailing down the numbers. And this is the one factor, ADME and toxicity prediction. This is something what I would like to put more emphasis. when you talk about drug discovery, there are two basically important types. One is structure based drug discovery, and other is ligand based drug discovery. So, in structure based drug discovery, you actually try to understand the receptor, their binding pocket, what are the characteristics of pocket. If you have known ligand, so you can uh, figure out what are the key important interactions or the amino acid of the proteins that you can target. Or if there isn't some additional pocket available which is existing ligands are not occupying, how you can modify the ligand or how you can design the drug so you can enhance the binding. Now, in ligand based drug discovery approach, say for example, you have uh, certain drugs. For an example, for epilepsy, you have levetiracetam, but most patients are not responding. So, if you have carbazepine or simply carbazepines, you want to design something new which is more potent, which can have less resistance. Then you think what are the characteristics of this active compound already, because if it is active, it is binding to the protein. Then you can try different uh, analogs of the pharmacophore. You can try to develop a SAR, and you can then screen and find more potent compound, more efficacious compound based on the ligand uh, optimization. So how AI handle chemical compounds? Now this is something like more interesting. I guess you will find actual insight how AI actually processing the compounds, how the softwares are actually analyzing the data. So they can whatever the command, whatever the program you want to see or you want to select, then AI is doing a job for you, and then there is an interface where you can analyze and your data output, and then you can even you know experimental design, you can plan accordingly. So for an example, this is a structure of a chemical compound. Now for this chemical compound, I want to screen a library. How I'm going to do that, or how this AI, how it will screen a multi-million compound library based on this structure. So these AI first generate a graph. So once the graph is generated, there are called nodes and edges in the AI terminology. So when I said nodes, so it will try to identify, the property of elements are like there are how many heterocycles, they are single bonded, double bonded, edges. Then they consider, okay, how many aromatic ring are there, alicyclic ring are there, heteroaromatic rings are there. They just put a filter. Then in this graph again applied based on the equivariance, and so this terminology I am trying to explain here in this slide: invariance and equivariance. For an example, this is the structure of coffee. So it this structure itself have the endogenous property of log s for minus 1.1. Now if this structure I rotated by 90 degree, does this property gonna change? No. Again, if I turn it 90 degree, is this property gonna change? No. So it is an invariance. That is the property of that ligand which is not changing if the structure rotates or it simply, for the AI I am talking about. Now, what is the equivariance? If the structure rotates like from the one side, are the other portion of the structure rotating by the same angle? For an example, so if I rotate this structure by 90 degree, all other are having the same impact. Yes, so then it is a equivariance. So in this screening parameter, then this equivariance invariance is applied, and then they filter the whole library, and then they give you certain kind of output. Secondly, this is a one more module that is called a diffusion module in the AI. Now consider this image. If you just want to get the very small portion of this image, the full image, it will give you the lot of noise. But if you want to again reassemble these noise together, it will give you the image, maybe closely related kind of thing through the AI. So this same principle is applied into the drug discovery. How? So for an example, that is called the de novo generation. So in this diagram, you can see in a protein, AI has identified what is actually there is a size, there is a specific dimension of this pocket. There are different kind of site points available where different characteristics are present. So AI just segregate all those characteristics together, and then for an example, this is a cluster of all the parameters, and then these parameters of the cluster parameter is applied to the structure. Now, based on this, whatever the compound matching this, they get forwarded again to the next level, and the best matching, the AI will give you the best matching as a output. So this is how, for a drug discovery, you can see AI is processing the compound. These are some literatures I'm going to show you for the drug discovery using artificial intelligence. This paper is specifically talking about designing dual targeting compound, like the one compound having activity on two different targets. So you can see in this diagram, the two different compounds, they align together like this. I'm talking about them is AI, and they had AI identified certain features. They identified, okay, hinge region, what are the features, which are the key amino acid interacting with what region, what specific fragment, and based on that, then into the this is a chemistry 42 another module. They have designed different hinge region scaffold solvent exposure groups, and try to design a different set of ligand, because now they merged two compound different compounds property altogether. So a novel compound has been designed. Then based on SAR optimization, if you can observe here, affinity, the activity value here, if initially it is 261 nanomolar, through the SAR optimization, the potency increased significantly with the binding affinity of 19 nanomole. So again, once this ligand is available, individually it get docked with the receptor. They observe what are the regions still you can have space to modify, and then they have placed another functional group on this position, where from 19 nanomole binding affinity, it again improved to 1.7 nanomolar binding affinity. Now, last parameter is DMPK, like drug metabolism and pharmacokinetic optimization, where you have to consider its solubility, you have to consider the metabolic stability as well, and then imparting all these qualities, still you can see there is a predicted and actual compound they synthesize is having still high potency. It is just like a nanomolar range with a single digit nanomolar range activity. They are highly potent, and they are promising candidate to be successful into the, at least I will say, preclinical. And then if they all parameters match into the preclinical evaluation, they will head towards clinical trial. This is another reference article talking about using AI into the designing and synthesizing some of uh, novel compounds. The article itself, like they mention how rapidly they get into the drug candidates, and how they got novel enumeration, prioritizing, and favorable drug like properties. If you want to read more on this, so there is a textbook, phytochemistry and computational tools and database in drug discovery. I have written one of the chapter in this book. So there are, I also mention actually machine learning, artificial intelligence, whole process in detail for the natural product based drug discovery, considering actually the pharmacy in a different branch wise. I just want to highlight some of, like, key areas when you consider the pharma chemistry, because that is something which use more frequently. They use for the docking, QSAR, synthesis planning. These researchers can use. I will say pharmacognosy. It is more useful to identify pharmacophore by having ability of high throughput virtual screening. So you can, because at least for pharmacognosy, stereoselective compounds or isomer specific chiralities are very important into the natural product. You can retain into the high throughput screening, and you can design different pharmacophore. You can then collaborate with chemists, or say for example, if you have certain compound screen for a target, you can collaborate with pharmacologist. Pharmacologist can utilize artificial intelligence into identifying a biomarkers. So typically, based on the expression value, maybe downstream signaling pathways, they can identify, okay, this specific protein is overexpressed in certain disease. So can we put this as a marker? Is it valid marker for this disease? And for example, they can use different data for the pathogenesis, and then they can, you know, early detection or the diagnosis wise, pharmacologist can develop some protocols. In a pharmaceutics, this is very important and very emerging right now. Formulation design. So specifically, you can, I think previous section I already touched on the crystal structure, co-former, co-crystallization. So all these you can, CCDC database she was talking about. So you can screen or you can preliminarily filter few of the co-formers from the available literature and database, and then you can select some of most suitable co-former for your synthesis. And crystal structure prediction, this is also very interesting and emerging. Many of the softwares giving these module right now, I think most are they are paid, but this is something very interesting if you are trying to design something new formulation, maybe co-crystals to improve some of the pharmacokinetic, because not attaining specific potency or the activity or the target binding is important. The most important to having pharmacokinetic parameters which are very optimum for the delivery, dosage, uh, form selection. And that's why, because still now we preferred oral one, that is more common in the market as well. And pharma industry also prefer the oral because considering the cost of manufacturing and all. So I think now I'm just trying to cover, giving you the brief background of AI in drug discovery. My focus is for target identification and drug discovery right now. This is taking, multiple horizons into the pharma industry. You have some industry experts within you right now, so they will gonna touch on a schedule I saw. They are talking more into the different protocols. But here are certain resources if you want to take a note of it. uh, they are some of available as a free, some are available with an academic license, you can request them. Swissdoc, all these are freely available. I think Edison is, it is also available freely. I exactly current status even I am not sure for few of them, because initially once they develop, they make it freely accessible. Over the period of time, once they get more users, they take it out of, the free access and make it license version. But still most of them are at least academic, like bolts 2, I think this is still free. And finally, I would like to acknowledge my university at the Creighton University, my mentor, Elton Narth. You can find he's a author into the FOES Principle of Medicinal Chemistry. I would like to thank Dr. Khedekar. Because of him, actually, whatever I am today, he is the foundation. He is actually the person who's uh, helped me in my foundation of the career. Dr. Rupesh Chikle, these two person actually always there with me. They are, my godfather, I will say. They are giving me proper input, suggestions whenever I am confused, or if I could not find a way. Uh, I used to talk to them. And finally, most important, I would like to thank Nagpur College of Pharmacy for inviting me for on this occasion, giving me a chance to talk with these active audience. And thank you very much. But I just want to mention one of the quote here. What all of us have to do is to make sure we are using AI in a way that it is for the benefit of humanity, not to the detriment of humanity. This is what exactly applicable to the pharmacist. And I think with that note, uh, I'm just ending my session. I think I try to wrap up within a time frame, but thank you very much. Thank you all. I'm getting some encouraging comments right now. Thank you very much. I hope you like this one.
Hello. On behalf of organizing committee, I sincerely thank Dr. Amit Pan for delivering a wonderful and thoughtful, knowledgeable talk on AI-driven drug discovery. Now, the session is open for question answer session on both the modes.
Uh,
Anyone
if like really someone has some of like key concern right now, I'm trying to address those, but because of the time constraint, and I think it's almost uh, 1:30 here in the night, and I have again still I have to attend another program right now. But if still you have something, I can quickly answer.
if anyone having question, they can ask via chat box also. Sir will answer them.
Or even like if any time in
I think
between
no one has
any time.
having any question related to drug discovery. Once again, I am thankful to Dr. Amit Pan for sharing his valuable thought on AI-driven drug discovery. Thank you very much.
Amit,
Thank you.
Khedekar here.
Yes, sir.
Because
Yes.
nobody is asking questions, that means they have understand everything or they have understand nothing.
Absolutely. I guess so because AI, the the audience who is not with the field in the technology, and I am a person who is not related to technology. I am just user, and these are the future users. So I am doubting myself like how much I can actually deliver this one to all of you. But hopefully, anytime if you find something wherever in my capabilities I can help you, I would like to help all of you. And obviously, you can have my contact information through Dr. Khedekar. He is actually my mentor into my PhD research, which I completed into UDPS.
Now here,
Thank
one
you.
thing I want to point out that Dr. Amit is one of my good student, very hard working, and today morning some technical issues were there and I requested him that we are shifting your lecture on second sequence, so immediately he accepted our request. Thanks for that. And in
Anytime for
US
you,
now
sir.
it is 1:30. And
Yeah, in the
another thing
night.
I want to mention here that last few days back, Amit's father expired. So I came to know that news. But in spite of that, he has accepted our invitation. And today, he informed me that the Tervi function is going on in his village, Chikhli, Buldana. But still, he has skipped that function and he has joined us. So Amit, I'm very much proud of you and I convey thanks on behalf of Nagpur College of Pharmacy and our principal and our team. Thank you very much.
Thank you, sir. So, and excuse me, I'm leaving the session right now because I would interested to attend the whole, but time is not allowing. But thank you. Have a good day. Enjoy the sessions.
Thank you so much, sir. Now we are moving towards our third invited talk, that is by Mr. Vikram Singh Suryavanshi, sir. He's a computational chemist, SilicoScientia Private Limited, Bangalore. And he will present on in silico crystal structure prediction, a paradigm shift in structural biology. And for this session, I would like to invite Dr. Sachin M. Hiradeve, sir, to chair this session. Sir, please kindly introduce the speaker with us.
Thank you, madam, for inviting me to introduce our eminent speaker, Vikram Singh Suryavanshi. Saraswati, respected Principal Sir, Dr. Khedikar Sir, my dear colleague and participants. It is my privilege to introduce our speaker, Mr. Suryavanshi. Suryavanshi sir is a junior computational chemist at SilicoScientia Private Limited. Recently, he completed his M.Pharm in pharmaceutical chemistry under the guidance of Dr. Khedikar sir. And his achievement, he qualified GPAT 2021 and qualified Nagpur JEE 2021, All India rank 804. He have also expertise in drug discovery tools and AI/ML based drug discovery. In very short span of time, he published many papers, research publication in various reputed journals and attended many training and workshops. So with this, I would like to hand over the session
Called football, not soccer.
I would like to invite Thakre sir to welcome our guest. Let's begin.
बहुत से बहुत से
Thank you, sir. Now I would like to hand over the session to Mr. Suryavanshi.
नाही मी नाव ठेव गेला आता गणेश सोडून तिला वाटलं की हिने गणेश आहे का काय मध्ये म्हणलं आता काय माझं लक्ष नव्हतं पुढं आलं माझं लक्ष गेलं तिकडं नाहीतर मी तिथेच थांबून तुला विचारलं तेच आपलं मगाचं बोलणं अरे येडे मगाचं बोलणं अरे ती बघितली पण मला
Okay, so good afternoon everyone. So I am here today to introduce about the crystal structure prediction, that is CSP, using SilicoCSP. Okay, so what are the crystal structures and all information regarding the CSP utilized in the pharmaceutical company in the formulation and along with the R&D. So we are going to introduce all the procedure here. Then first of all about what are the crystal structures. So crystal structures are the solid with highly order repeating arrangement of atoms, ions or molecule in the 3D lattice. So we want to first understanding what are the crystal structures. So in our daily day-to-day life, there are so many examples are available that we are use in your day-to-day life that are in the crystal form. So crystal form are nothing but the solid compounds with the repeating unit of the atoms or molecules in their 3D structures. Okay, like example table salt. Okay, normal table salt we are utilize in our daily routine. It is in the cubic shape. Then the sucrose, it is also available in the monoclinic form of the crystal structure. Then ice is the water molecules crystal structure in the hexagonal. Then the diamond is also the crystalline structure as a tetragonal geometry. So these are all the our day-to-day life daily routine compound that are available in the crystal. Apart from that, metal compounds are also available in the crystal structure like silicon. So many metals are available and that utilized in the various type of electrical instrument like our LCD, LED and so many on. So applications of these crystal structures. So in that so many types of applications are available for the crystal structure. Including porosity gas storage application like some department wants to fill the gas for the some explosion factory something. So there we want some type of the gases for their porosity calculation and the gas storage. So that particular criteria CSP utilize this particular section of porous and gas storage. Then in the second era, energetic material are prepared using the crystal structures, same as metal compound utilized in the power section or the explosive compounds. So in this era, CSP prediction is very essential because some are the very explosive in nature, some compound are very explosive in nature. So we are going to first study all the details of the explosive compound previously in silico using crystal structure prediction. Then after that we will go for synthesize in the laboratory for avoiding the major blast or major explosiveness in the laboratory. Then third one is the electrical properties utilization in the electric field like silicon computer chips, then gallium arsenic LEDs, then solar cells, and apart from that this crystal structure prediction also utilized for calculation of so many types of chemical properties like compounds reactivity and along with that their interaction with the different type of co-crystal formation compounds. And lastly, the application in our pharmaceutical properties. We are going to design some compound which are available as a low solubility, viability, and another type of the physicochemical property. So we want to design active pharmaceutical ingredient with the that compound which are increase the solubility of that active pharmaceutical ingredient. So we want to merge API and co-former togetherly for the increasement of the solubility and other parameters of the this formulation. So we will demonstrate whole tool in the second half. So these are all about the applications of crystal structure. Then after that, what are the experimental method utilized for the crystal structure? Just this is a short info about how many types of the experimental methods are utilized in the crystal structure. So it is a very tedious and slow process to perform all the techniques in laboratory or something. So first is slow evaporation, then second is a vapor diffusion, slow cooling, then small molecule crystallization, then liquid-liquid diffusion, seeding, crystallization. Crystallization is very well-known technique. We will utilize generally in our lab experimental techniques. So these are all the experimental methods for generation of crystal in our synthesis process or last purification technique. Okay. So in this process, we want some solvent compound that is miscible in our drug compound, and that solvent we will generate the crystal after some time or within 1 day or within 2 or 3 days according to their time. So these are all the techniques we are utilized for the experimentally techniques for the crystal generation. Okay. Then next slide. Then after that, this whole process takes so many types of duration. So total experimental timeline for the crystal generation takes so long time. Like suppose you can see here, first step is crystallization screening. It depends upon solvent, temperature, and supersaturation state. So definitely it is comes as a very long process. It will complete in day or sometime week. Another step is a crystal growth. So it is also very time consuming, and it takes around extended period of the time for the crystal growth. And lastly, structural determination of this crystal structure using XRD or single crystal structure. So these are all the experimental technique takes so long time for the determination of whole the process and growing of the crystal structure in our laboratory or any research labs. So for this whole experimental timeline, we are going to introduce in silico method with all the drawbacks overcoming using in silico method of CSP. So first we will go through the difficulty of challenges of experimental crystal structure. It is very time consuming and lot of resources required for the large screening data set, and also the it traps lastly in the kinetic and thermodynamic techniques, this thermodynamic techniques calculation of energy that we have to calculate before synthesis method of crystal structure. So it takes some fault in the unstable metastable form of crystal structure. So this is a drawback of experimental technique. Then polymorph generation uncertainty, and lastly scenario of lab form means whether it is available, scenario available for the lab synthesis or something. So overall, to overcome all the drawbacks of the experimental crystal generation, we are here to introduce our SilicoCSP tool that is totally depend upon in silico crystal structure prediction that overcome the experimental and the crystal structure generation process. So for that, first, what is the in silico method? In silico method are the most stable 3D generation structure using the molecular structure. Then it is widely used in the drug discovery for the APIs and co-former. API is active pharmaceutical drug, and co-former are the excipient that is utilized for the generation of complex with API. In the general in silico crystal structure, basic workflow is there like molecular structure of API and co-former molecule. So we have to generate their structures using AI based some machine learning techniques and some tools. Then after that we have to optimize structure using some GNN model or AI based model, and we calculate the energies of all the generated API plus co-crystal molecule compound, and we relax energy minimization structure for the calculation of the stability of this API and co-former formulation compound. And lastly, we rank this compound according to their energy activity and energy rank. Okay. Suppose lastly, then we can conclude as a stable crystal structure we predicted through in silico. Now we are going to synthesize in the laboratory. So from starting to it, suppose I taken 100 compound API plus co-former co-crystal molecule for generation of CS co-crystal. So out of 100, lastly, hardly 10 to 12 compound are find out as a stable. After all this process will go through the 100 molecules. Okay. So this is a basic workflow of in silico crystal structure prediction. We implemented in our tool. Then after that, why CSP is important, why crystal structure prediction is important. As we know, there are so many types of uncertainty and instability comes in the formulation in the synthesis laboratory or something. So to avoid a late stage risks and cost of this synthesis compound, so we have to first perform all the in silico study of this crystal structure prediction. So it is very important. Then lastly, it increases the solubility, dissolution rate and hygroscophicity of compound. And also it is experimentally less cost, and it is a very less time consuming. Okay. Then after that, so generally in pharmaceutical sciences and other era, there are three major role of crystal structure prediction available as application in pharmaceutical design, in the material science, in the molecular packing and the properties. Then about just short info about the case studies of CSP. There are 4 case studies are involved with this CSP in the pharmaceutical design. First is the MK8876 drug compound. So this drug compound CSP was successfully predicted and ranked the malic acid co-crystal co-former as a complex, and they generated co-crystal as an antiviral candidate through this in CSP in silico method. Then like galuniceptive, this drug molecule also had made so many types of the complexes, like 50 plus polymers are generated and ranked according to the thermodynamically their energy stability. And last two drugs, dalcetravic and the 4-amino hydrobenzoic acid. These are also treated in the in silico study for the determination of their polymer stability and all. So these are the 4 examples case study for the CSP in the pharmaceutical. Next one is there is one case study happened in 1998 regarding ritonavir drug. This drug has came in the market around summer 1998 in the semi-solid capsule form, Norvir semi-solid capsule form. But after some time in the same year, it showed uncertainty and instability in the their polymorphic form. So the market recalled back this semi-solid capsule of ritonavir and study about their polymorph characterization using different type of spectroscopy, XRD and all. And they generated 2 polymorph of this same ritonavir drug, stable form second and stable form first. So this first second form is a more supersaturated and more stable form which are comes again in the market after this case study of the ritonavir in summer 1998. So this is a case study performed and this after this tragedy of the ritonavir. So again a short quick introduction on CSP material science. In the material sciences, their CSP are available in different type of the workflow, like input molecule compound, which required then energy calculation using the lattice energy landscape calculation, then the predicted the stable crystal structure, then the energy ranking using the molecule. So here lowest energy is the most stable crystal molecule with their co-former with their API. So as much as lowest energy, it is the most stable compound. And lastly, this material science department CSP utilizes in the energetic material, our pharmaceutical and optoelectric devices. So lastly, after this whole work, we will go for the and all experimental CSP, included and designed this CSP. CSP is our design tool by our SilicoScientia Private Limited. So generally, some API drug molecular problem regarding pure solubility, viability, something. So we designed this tool that predict stable and metastable crystal form of any API molecule with their co-former compound. API molecule such as any paracetamol, any ritonavir, if they have very less solubility, so we have to just search about the co-former compound that are available in the market or something, and we want to make a one co-crystal molecule complex with that molecule with our SilicoCSP tool, and then after that we will analyze all the energy and the thermodynamic stability of co-crystal molecule, and then after that we are going to enable and optimize this CSP generated structure through the energetic values and all. So this is the role of CSP tool, and this is the interface of our crystal prediction tool. Then key features of our SilicoCSP tool, definitely it will eliminate the need of experimental crystallography, all timeline relation that required in experimental. So definitely it will eliminate the time and it will complete short time period for the crystal generation. Then it is AI based model tool and Python based platform that we generated for crystal prediction along with the co-former for the API crystal complex foundation for the improve their physicochemical property along with their adamant properties and all. Then it follows multiple steps which generated some workflow for calculation of for creation of stable CSP complex for API and the co-former. So first step is the crystal generation, then GNN energy calculation. It contains the calculation of energy of CSP compound in electron per volt atom, and lastly MD simulation of co-crystal generated compound, and lastly X-ray diffraction plot generation of that generated co-crystal complex. Data of their co-former, and it works on the GPU and CPU compatible core. So and it is a very short period of time the whole process complete. So it is very beneficial in silico method. So this is our just short quick review steps of our SilicoCSP tool. So in that same we have to create the crystal structure using AI Python based programming. We require molecule with co-former in the XYZ file format. This XYZ file format you can directly get from different type of databases like PubChem, or you can convert through different OBABL techniques and all. And second step is the calculation of energy using GNN based AI generative based model energy calculation, and we have to select top energetic co-crystal molecule of user given API and the co-former. And lastly, MD simulation of that top co-crystal compound, and calculation of their kinetic energy, potential energy, and total energy with each MD frame of for the 1 picosecond, 5 picosecond, 25 picosecond as per user required. And lastly, we are going to plot X-ray diffraction plot of that generated compound, and we have to download all the required file output files of this CSP workflow. So this is a general introduction about our workflow. We will see in this whole workflow demonstration in our second half session. Then same user required this XYZ file for the starting of this tool, drug and co-former. Then it will give the crystallographic information file of this generated API and co-former crystal structure. And then after that, same steps are here mentioned with their time. Crystal generation takes only 1 to 2 minutes for produce the multiple crystal structure with one API with multiple crystal structure. Like one API, suppose paracetamol with different co-former like uh, malic acid, citric acid, or 4-amino benzic acid. Suppose you given three co-former with one API paracetamol, so it will generate three co-crystal molecule with different co-former like paracetamol malic acid, paracetamol tartaric acid, and paracetamol 4-amino benzoic acid. So this step only takes around 1 to 2 minutes according to number of molecules. Then second step hardly take 5 to 10 minutes for the calculation of energy and relaxation of energy based co-crystal molecule. And lastly, MD simulation that takes 5 minutes for the 5 picosecond. So overall, this whole step complete within around 30 minutes max for 5 co-crystal molecule generation. According to molecule, the time varies. Then this is a result of our SilicoCSP tool. Suppose we given the XYZ file of ritonavir drug molecule along with saccharin co-former for the increment of their solubility and after synthesis parameter. So this will generate likewise crystal structure with their unit cell of triclinic unit cell, and it is a primitive type crystal structure. It will give all the information about lattice, its lattice type, phase group number, its length, ABC, and its angle alpha, beta, theta, and details it will give after this first step. Then it is a primitive means all the ligand and co-former compound complex are attached with corner of unit cell. So this generated we will use for the next step of the energy calculation. Then this is a result of our second step, that is energy calculation and MD simulation. So we calculated here the energy of per atom in electron volt. So it required directly CIF file of first step generated co-crystal molecule that definitely automated go for the energy calculation, and it will generate this type of CSV file including file name or name of compound, then energy per atom EV, and this dimension of the lattice information. So here as much as negative value are higher, the stable co-crystal molecule are available. Like you can see here minus 6.67. This second compound carbamazepine with gallic acid co-former with API carbamazepine. These compound are more stable with first compound ibuprofen nicotinamide that shows minus 6.20 energy per atom range. So we can rank this compound according to energy and all, and we can select top compound from this for the MD simulation also for our for the narrowing down the compound with co-former with API. Yes. Then third last step is the MD simulation. So we proceed for MD simulation of that selected compound in the second step. So it will generate this type of log file containing time in picosecond from 0 frame to the 1 picosecond frame, and its energy calculation, energy of total, energy of potential and kinetic. So potential total energy is a summation of potential and kinetic energy, and last is temperature. Every step temperature we have to here note it down, like somewhere temperature is higher, somewhere is lower. So overall the interaction of MD simulation, molecular dynamics simulation of that API compound with different co-former we have to study it in this MD simulation. So this is the third step of our Silico CSP workflow. Then interpretation of MD results. So MD interpretation of second step result, like energy per atom. Same as I mentioned, lower energy per atom means crystal structure more stable as comparing different form of the same compound. Like here I use API, one drug molecule with different co-former molecule. Okay, so we can rank according to energy in this step of the second step. Then in the MD simulation, we have to also interpret MD simulation with changing of time like 1 picosecond, 5 picosecond, and then 20 picosecond. So after that, we have to calculate the distance between first frame and the last frame for the determination of the stability of compound. And according to that, the lower difference is the higher stability of this co-crystal molecule. Like with carbamazepine, cytosine API is a more stability showing in the in silico MD simulation with 4.79 in the 1 picosecond, 10.72 in the 5 picosecond, and 10.61 in the 20 picosecond. So these are all the comparative study in the 1 picosecond, 5 picosecond, 20 picosecond. So you can do this type of study in our silico CSP. If researcher want to study comparative study, also researcher can like this way. These are the MD parameters that we are utilize in our Silico CSP tool, like temperature, atoms, their time in steps, then trajectory, output file name. All the details will provided in the Silico CSP tool. Last step is generation of the XRT plot of that generated co-crystal molecule as a final four-step compound. So this is a graph of favipiravir and nicotinamide compound that generated by our Silico CSP tool. So after lastly, we can demonstrate and we can interpret this graph using 2 theta angle declination and normalized intensity number ratio of this XRD plot. So here higher peak, narrow peak shows the crystallinity of the compound. So in that graph around 10 to 20 theta angle, this compound is showing the higher crystallinity in its nature, and after the 30 40 later on, it will show the amorphous in nature. So lower peak and broad peak shows the amorphous in nature, and the normal narrow peak shows the crystalline in nature. So we can interpret the nature of the compound lastly through this XRT graph generation also. So this is all overall our tool interpretation. It's running and all. So overall output of first. So overall output of Silico CSP, it will give first the crystal structure of any API and drug co-former molecule like agomelatine API and saccharin is a co-former. Then in the second step, it will provide the energy per atom calculation. So here we have to rank the compound according to energy. And then in the third step, we transfer this compound to MD simulation energetics. And lastly, we selected some compound as a stable compound for the generation of the XRD plot for the crystal structure. These are the four outcomes that we are going to introduce in the our Silico CSP tool. Then perform short validation and benchmarking for the CSP tool. According to the energy, we interpreted the basic range of this molecule like 0.01 less than electron per volt are the good stability compound, and apart from that like 0.01, 0.02 in between 0.03, 0.4, this is acceptable range, and more than 0.05, it is very insignificant energy difference significant, so it is unstable co-crystal molecule. So by this chart you can interpret the stability of compound, and from that we calculated the energy distance of this whole co-crystal molecule like energy. This calculated we done on the 16 experimental crystal structure we extracted from CCDC database, and we calculated we generated same crystal structure of 16 experimental crystal structure from our Silico CSP, and that 16 compounds data with our generated compound data we compare and calculate the energy distances and in its stability. So outcome of our validation, we are total out of 16 compound, 11 compound are in the good range. They show the stability through overall the steps. To 68 to 75% are the compounds are in our side in the stability. So it shows the 68.75% outcome with this all the ranges. And also we tested with the similarities calculation of our generated XRD plot and the already established CCDC compounds of the co-crystal molecule. Next. So lastly, finally, our SilicoCSP that reduces the experimental workload and development type, and it will avoid the risk of any synthesize step, the failure risk in the after the synthesis process. So researcher want to do a before synthesis any CSP related research. So this SilicoCSP tool definitely helpful. Then so many types of the challenges are involved, but that all challenges we are overcome in the SilicoCSP. So these are all the references we utilize for the study and all. So thank you so much for your kind attention, and thank you so much.
Now the session is open for question and answer.
Hello, sir. My name is Shakir Ali, and can we access this tool, Silico CSP?
On the behalf of organizing committee of Nagpur College of Pharmacy, it is my privilege to summarize this session in silico crystal structure prediction. Sir has elaborated the crystal structures, application of crystal structure, and experimental methods for the crystal generations. I would like to thank to our esteemed speaker for sharing his valuable knowledge, experience, and insights. The clarity of presentation and relevance of topic have truly enhanced our understanding. So with this, I would like to declare the session, and the session is concluded here. Thank you.
Hello, sir.
Thank
Am
you
I
all the
audible?
delegates for your kind patience. And now it's time for lunch. And before that, I would like to announce that sharply at 2, we will start with our remaining session. And all the delegates are requested to please fill the feedback form available in your kit, and please submit it while collecting your certificates. Now you can proceed for the lunch. Thank you. Lunch is in the canteen, please. Lunch is in the YCC canteen.
If you keep starting strong and then suddenly losing momentum, it is not
ऍक्च्युअल मॅमचं केव्हा येतं?
अठराची डेट आहे पण 18ची डेट आहे पण म्हणजे 20 तारखेला रेडी नाही म्हणून त्यांना शकल 19th ला सुट्टीचे आम्हाला मागे म्हटलं होतं बघा सुट्टी राहील पण एकदाच सेकंड कसं करायला लागेल मॅडम सांगू तर किती सब्जेक्ट आता चारच यांच्याकडे प्रॅक्टिकल येते का फर्स्ट इयर अरे ते वेळ जागा सोडली आहे 17ची खोली का 17ची खोली आहे हा 16 आणि नम्रता त्यांच्यासोबत नम्रता माझा डिग्री आहे अमरावती गव्हर्नमेंट. माझा नंबर तिकडे गव्हर्नमेंट कॉलेज हा बाहेर ऍक्च्युली माझं एज्युकेशन बाहेरच झालेलं that is academic education हो त्यांना काहीच टेन्शन नाही, त्यांना माहिती सगळंच करून देतात. खूप चांगली आहे. डिझॉल्व होत की नाही माहिती, डिझॉल्व होतात झाडाला काय बाकीचे मग युनिव्हर्सिटीने एक्सेप्ट केलं पाहिजे ना एवढे कॉलेजेस बाकी मला वाटेल माझे माझं नेक्स्ट बॅग आहे ना त्याच्यात फॅमिली
आवाज येत नाहीये. Hello, anyone is there? Hello. Anyone is there? Voice is not audible.
So your jobs are already like this. So with the time being, you have to adapt a new technology. In 90s, when the computer came in India, a lot of people actually opposed it. No computer will come here. If the computer is coming, the people lose the job. The computer coming with computer, a lot of jobs created. And similarly now, some people are worried about that is yes, if the job AI is coming, the people will lose the job. Now, this is not true. This is not true. So if you have a skill, no one can match you, and the entire world is yours, definitely. So now the question is that, is the AI threat or opportunity? So I mentioned, yes, it is not a threat, it is opportunity. And we are fortunate in this age, that is the AI cure. So we can, if we adopt it, the lot of opportunities here. So, thing is that we are trying to replace human, but human with AI will definitely replace without human without AI. Okay. So if you don't, it is very much true that AI never, it is not possible for AI to replace the human. But if you are not skilled in the AI, those people that know the AI replace them. So the technique you have to change accordingly. Now I am going to just briefly explain this thing. So in a drug discovery field, the computer drug designing, these are the different approaches here, and the most of the revenue is coming in the particular company. So this amount of revenue coming from this kind of approach like computer drug design, and this is a new technique for the But even if you projected that after few years, maybe couple of years, the scenario will change. What will change? That is the revenue, the most of the revenue comes with the advanced application of AI in the drug discovery field. And the client is pharmaceutical company, research center, battery company, or institution, or CRO. And their client will be like, their job will be like drug discovery, or end-to-end discovery, or multi-omic, etc., synthetic, etc. And these are some outcomes. So if this actually changes that, if I go back a little bit behind, that normal, the traditional drug discovery is replaced by the computer drug designing, and now the computer drug designing is replaced by the AI drug discovery.
Hello, sir. Oh, hello. Hello. Oh.
So I think this is the success story, kind of short question someone asked actually what is going the success. Yes, so these are some molecules are here some companies who can clinical trials with the help of AI and machine learning things. So this is long. So these are the molecules you can add here to maybe the list is more long. So these molecules are different skills of clinical trials and they can do the AI-based drug discovery approach. So this is actually, yes, this has a lot of issues also, like problems also, like data, so good data, bad data. If you have a good data, then definitely you are, you have a good knowledge, and that productivity will definitely good. But also the knowledge, you have to understand your data, you have to understand the approach you would like to apply, that is also challenge. Business model, the IP, the data sharing, distribution, these two things are very complicated things, especially in the academic institution. Because data you have to share, okay, and also you have to distribute the data. So that actually only possible we have the centralized system like cloud-based system or like that. So now the cloud is getting very popular in the market. What is the cloud? That is some server is present somewhere else, like UK, US, or somewhere else, and I'm working from there. So everything is running. Just upload the data here from there. Everything is running there. That is a cloud. So cloud is one cloud is very much important because first it is safer in storage, real-time teamwork, and easy data access and fast and disclosure. Because suppose I would like to do some expensive drug discovery project. So I need a good server. I have money. I'm a very rich person, suppose. I am not actually, but suppose I am a rich person. So I invest 20 lakhs for a computer. I purchased it. I installed it in my system. To maintain that, I have to hire another person. Because it is not possible. It is not my cup of tea to maintain the server regularly. So I have to hire the technician. So I have to pay maybe 60,000, 70,000, 80,000, 1 lakh per month. That is another. If server is down, either my system down, I have to hire some hardware. I have to ask for hardware to come down. So these actually complicated there. So if you have a cloud, everything is automatic. Dedicated team is there, 24/7 available. Depending upon suppose I have a work. Suppose I submitted some calculation. First 1 hour I need 30 CPUs. So it will use 30 CPUs. Suppose in next 1 hour the calculation is increased, I need 120 CPUs. So automatically 120 CPUs will assign. I know that okay, my computer is only 48 CPUs, how can I manage? Not like that, but automatically do it. Again, suppose the third president, next stage, it needs 48, automatically shrinks. So this is a very beauty thing of this year. Automatically the RAM will increase. So that's why the cloud is very much popular. And you will be surprised that 83%, about 83% pharmaceutical companies are using the cloud. They are not using on the local server. Due to this all audit. Okay. Now the another thing, the SaaS licensing, software as service. This is also getting popularity. Why? And I told you, if I go for some the very well-known software, Biovia, very well-known software, Precet, very well-known software. So first of all, if I buy those software, I have to install in my local system. So that is the problem that you have to buy this server, you have to hire a technical team, and you have to install. That is fine. I did it. But it is software installed. Suppose tomorrow some technical problem is there in the software. So what you have to write email to the storage or Biovia. They will check it. They will come with the annual. So this one whole 1 week you have no job. So that's why people actually software as a service. That means the product will run in the cloud. You will give the license. For one license, two licenses, three licenses, you have to just open your laptop to access that software from here. If anything is problematic, the cloud will check it. The technical team automatically check it. So that is called a SaaS software as a service. Now, this is actually gaining the very popularity. Because company or organizations don't bother about this, because others to maintain all these things, software is crashed, software is going to reinstall, the people will come. So that's why people are moving on this. So this is around 31% jump in the SaaS based software application. So, keeping all these things, that is a easy use of the cloud as well as a popularity of the SaaS licensing, SilicoScientia, they developed their flagship software in SilicoXplore, and this SilicoXplore is a completely SaaS-based, cloud-based tool. So maybe after this session, we'll see the demonstration of the tools. So it is a powerful cloud-based tool, definitely integrated platform for the entire statistical tool, and it is AI-based and physics-based combination tools. Some tools are AI-based, some are physics-based, and definitely it is a correlation range from here to region. So these are some packages. I think they will explain, they will show you how these packages are actually working right now. So CA255 peptide design, protein modeling, to see how the mutation will impact the stability of the protein, molecule developed in new molecule generation, novel molecule. This is the screening, machine learning process screening of the new number of molecule, QSR modeling, method prediction, similarity searching based on input molecule. Thousands to millions of molecules can convert in a different file format. This is the problem for features extraction. This is a composition generation, and this is the packet identification. So these are the two packages are there. Like document tools are there. Now four tools are there. There is Liganda, Vina, Doc, and DeepDoc. There is a metal docking also there. You can do the metal docking. Those people are doing docking, they know how difficult to dock the metal compounds. That also actually solved it. Interaction here, power kinetics, toxicity, chemical analysis, and here the DFT calculation, and here the molecular dynamics simulation. Because molecular dynamics simulation in Chroma, it is very tedious job. So we have developed an interface that you just upload a protein and ligand, and all things will take the cloud. So these are some metal backgrounds to so definitely, so as I said, this is a complete drug discovery pipeline. So suppose you are doing some drug discovery project. Some work can be done some software, and some other work you have to do some other software. So it is distributed. So that actually you can manage here the entire thing in a single platform. There's a wall in one platform. So this is actually the demo, but I'm not showing demo right now because our team will show hands-on the demo right now. So this video will how easy to draw and speak quickly. Yes, this is a very good cloud-based tool using physics and AI-based methods. So this is a case study. One of the case study, I think around 78 non-molecules are taken, around 11,000 Inaugural molecule is generated, and we spoke of similarity screening, molecular profiling, then pharmacokinetics, affinity calculation, flow refinement, solid power dynamics, and free calculation that's performed. And these are the data. And the methodology is submitted for publication. This is another case study. So here, I think we consider around 1.4 million compound percentage researching, and then we use silico scaling, proxy, SilicoXplore, all these things, and then finally we finalize the 6 molecules in this case study. So that I'm trying to explain here. So this We developed this tool and already we have tested with a huge number of data, and that is our severe accident. So here you can see these are some research papers we have published in the last 2 years using the SilicoXplore. So some before writing the SilicoXplore, some after SilicoXplore, but we have used the same code to get running behind it. So these all the papers we have published using this SilicoXplore. So in our case, suppose we are doing the client work, we are doing the development as well as the research work. So we are a very small team, 10, 12 people over here. We can manage the last 2 years, we can publish more than 30. There is a high impact journal, the Q1, Q2, Q3. So that is a skill that is, if you have such kind of the tools, so it is easy to do the data analysis and it will be publication or your research will be get momentum. So just in a quickly I will introduce my team. So I will need a personal product and personal journey. We can together to find out that when we discuss it, we found that yes, we are going to have people, those are chemists or pharmacists, they want to do the large chemical space exploration, but they don't have the expertise, they don't have the Python screening or programming screening. So for that reason, actually, we have started this venture and we have developed this tool. So these are the national international collaboration. We are working South University, Mr. Rasta University, and University of Nigeria. So we are a company we are working for. So these are the clients and products that we are actively working right now. So thank you so much for your presence. Okay, I think now we are bored about AI since morning for this AI. So I'm sorry for that, and thank you so much for your presence.
Thank you so much, sir. It's my pride and privilege to summarize the session. Dr. Atul sir has explained regarding the artificial intelligence and machine learning challenges in computational drug discovery, AI in pharmaceutical development and challenges of AI in drug discovery, and many aspects regarding artificial intelligence and machine learning. So I express my deepest sense of gratitude towards Dr. Atul sir, Head of Science and Director
SilicoScientia, Bangalore, for his mesmerizing valuable guidance, which will really help us to accomplish the ambitions of my students and live to a high potential. Thank you so much, sir.
Thank you so much.
Excuse me, sir. May I have
Yes, sure.
Because he did a nice presentation.
Mic band. Mic band.
Hello.
Yeah.
Sir, I will discuss about the study of the AI and its advantages. Especially
The main limitation is the data. Because in some cases data are available, in some cases data are not available, and what to do. Because if you say there is no data, so there is a lot of limitation in the to purchase 4 GPUs. So this is another big problem. But ultimately, the success is there.
it has extended its branches only in the drug repurposing and like that.
Not exactly.
In drug repurposing, the AI is helpful, but in other sectors like already we are having an advanced software like computer design, many software, Scott Intel, Life Sciences, or many more Autodoc and some software also. So what is the
I'm talking here. Suppose you have Schrodinger. I'm asking the question. How many compounds you dock at a time in a machine? Maybe 1 lakh molecule. 1 lakh molecule. I was saying that you are very excellent at 2 lakhs molecule, 5 lakhs molecule. I have suppose 50 crore molecules. What you will do? That the AI will help. So I want to explore this 50 crore molecule. So what actually we do through AI? AI actually it is not the replacement of the existing tools. It is the helping the existing tool. because the Schrodinger, Biobuak, they are physics-based tool. There is no replacement at all. Physics-based is a standard tool already established. So AI actually help this tool. So suppose in a physics-based tool, the Schrodinger you can manage 5 lakhs compound. So I have a 50 crore compound. So what I will do, I will dock 50,000 molecules in a Schrodinger. I dock 50,000 molecules. I can't dock 50 crore. Am I right? So once I dock the 50,000 molecules, I have the molecule, I have the binding energy. I'll use the machine learning to develop the model from this data. Once I got the machine learning model, the remaining molecule I predict the binding affinity. Though I can't do in the Schrodinger. So that way AI actually helping you, assisting it. So it is actually in a physics-based tools, whatever tools here, there are some boundaries there. The AI actually break the boundary. You can go the universe to accept as much as data you want to explore. That is actually beauty of the AI. Okay. Any other question? Anybody? Okay. Thank you so much. Hello.
To proceeding our next session, I'd like to invite Dr. Pramod Khedekar sir on stage, Mr. Kunal Saire and Ms. Suhani Gawane, please take your place on the dais. I request Mane ma'am to please present the token of love to Ms. Suhani Gawane. I request Khedekar sir to please present the token of love to Mr. Kunal Saire. The morning session provides us an insightful discussion on artificial intelligence and its transformative role in drug discovery. Now we move towards the most exciting and practical segment of today's program, the hands-on demonstration and advancement application of the session. To begin with the session, we have live demonstration on in silico crystal structure prediction. It is my privilege to invite our resource person, Mr. Vikram Singh Suryavanshi, a computational chemist, and he is sir is serving as a junior computational chemist at SilicoScientia Private Limited, and Mr. Kunal Saire. So they will guide us upon through the practical aspects of computational drug discovery using advancement in silico tools. I kindly request you to please take the session and enlighten our participants.
और चाहिए क्या? माइंड स्क्रीन शेयर की क्या आपने? हाँ एक ही स्क्रीन का चलेगा। क्रोम है ना, क्रोम का कर लेना आप। दिख जाएगा पूरा। मुझे मिनिमम हेलो 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 हेलो। हाँ सर एक मिनट सुहा मोबाइल नहीं, मेरा मोबाइल सुहानी के पास लगेगा। हाँ मोबाइल चाहिए। हो गया, कनेक्ट हो गया सब। मोबाइल में वो ये है ना, लिस्ट है वो देखना है मुझे।
इसी को करना जो चल रहा है, जो सर कर रहे हैं बस उसी को स्क्रीन करते रहो। स्क्रीन शेयरिंग लगा
झालं सर ते वर्क नाही झालं सर एक
सेकंड बस इसको डुप्लीकेट कर लेते एक्सटेंडेड की जगह, नहीं तो ये वाला वहाँ शो नहीं होगा जो आप यहाँ करेंगे।
ये नहीं होगा क्यों?
एक्सटेंडेड स्क्रीन होगा,
तो करो ना जल्दी।
अरे उसका शॉर्टकट होता है ना, उसको लगाओ ना। फोन कहाँ है बंदा?
वाला की बोलो करने उसको बना होगा शॉर्टकट वायरलेस।
स्क्रीन शेयर करने से नहीं दिखेगा क्या? आपका मेल आईडी क्या बोला था? जल्दी बोलो।
12 रूपम 9
12
रूपम
AM
91@gmail.com।
ये लिंक ओपन करनी है। ये लिंक जो मैंने भेजी ओपन करना है गूगल पे।
नहीं हुआ, फिर से कर, विंडोज की दबाए करो।
सर वो स्क्रीन शेयर मतलब दिख नहीं रहा है स्क्रीन पे, वो स्क्रीन शेयर वाला ऑप्शन हो गया।
जो पहले आया था ब्राउज़र में ओपन हो रहा है।
हाँ मतलब वो यहाँ पे हम ओपन कर रहे हैं लेकिन उसमें कुछ दिख नहीं रहा है वहाँ पे स्क्रीन शेयर में।
जो पहले वहाँ पे ओपन करो ना, अलग स्क्रीन पे कर बोलना, वायरलेस तो है ही ना अपने पास। अब माउस भी है तो वो भी दोनों है, तो वायरलेस चीज करना।
मैंने अभी आपको मेल भेजा, वो कॉपी करके गूगल पे ऐड करो।
वो वाला काम नहीं कर।
लिंक भेजे मैंने अभी।
टेक्निकल पेपर तो नहीं है जो वो सब करना टेक्निकल। अरे वाह
नेट तो है
टेक्निकल टीम में नहीं आता। रोशन सर अरे
इसको ऑन फिलहाल।
Okay, I think some technical issues going on. So in between, if you have any doubt or question, कुछ भी ऐसा नहीं है कि आपको इसी पर डिस्कस करना है. You can ask about job opportunity, कितना सैलरी मिल सकता है, क्या पढ़ना चाहिए, anything you can ask actually. कुछ क्वेश्चन नहीं आता दिमाग में कि
ये लिंक कॉपी करके गूगल पे डाल ये नो कर दिया। हाँ
डुप्लीकेट स्क्रीन करना है, डुप्लीकेट में सेकंड स्क्रीन डुप्लीकेट ओपन नहीं हो रहा, वो नहीं करवा रहे सर, जो भी होगा
ये नहीं वर्क करता है, अभी क्लिक हो रहा है। क्लिक भी नहीं हो रहा है। 4774 अभी हो रहा कुछ।
अभी ऑनलाइन कोई है क्या? ऑनलाइन कोई है?
तो वही इशू हो रहा है सर, ऑनलाइन दिखा नहीं पा रहे। तो ये वाला होगा तो हो जाएगा ना सक्सेस, चलेगा। ये अभी चल रहा था थोड़ी देर पहले। पहले ही हो रहा। तो नहीं होता है ना, पासवर्ड क्या है?
1 2 3 4 5 6 7 ट्राई करें।
एक बार इससे ट्राई करते क्या, अल्ट्रा व्यूवर से?
हाँ अल्ट्रा व्यूवर से।
Protein modeling, proteins active site identification, then after that molecular docking, admit properties prediction and all. So we will see one by one demonstration of each tool. So first I will start with CSP. This is a tool. I already given the seminar on this in the first half of this conference. So now this is the interface of crystal structure prediction. So as a first of all, user need to prepare job ID for this execution of the crystal structure prediction. So you have to first create a job ID with name of any name or any kind of alphabet here. So you have to click on this icon, create new tab option. Okay. Now you can see here your job ID is now created. You can note down this job ID for further fetching of your result after completion of the CSP protocol. Okay. So in this there are 3 steps. First is the structure generation, co-crystal molecule generation step. Second is a structural relaxation step, means calculation of energy according to GN model of algorithm, AI based algorithm. and third is the molecular dynamic simulation of generated co-crystal molecule. Okay. So you can see here first you need to upload the API molecule in the file format of XYZ file format. You require XYZ file format for this. So just you have to click on it, click to upload, and go particular directory where you have saved your all the data regarding the CSP. Okay. So you go to first folder of that for the uploading the XYZ file of the API. Suppose I am going to this API folder. Okay. So now select any one API molecule. Suppose I am taking agomelatine drug. Okay. This agomelatine drug is an anticonvulsant drug. We require XYZ file of it. So this XYZ file of you can convert through different type of the tools are available. Our SilicoXplore have given the tool Molcon. It is a tool for the conversion of one file format to another file format. Suppose you require convert smileys to XYZ file or SDF to XYZ file. So you can use that Molcon for conversion. After conversion, you just click on it agomelatine drug. Okay. Yeah, 1 minute. Connected? फिर से क्लोज करें करूँगा. कनेक्ट यहाँ से कट करके सर वो फिर से डिस्कनेक्ट हो गया अल्ट्रा व्यूअर यहाँ से डिस्कनेक्ट हो गया तो वो दिखेगा नहीं उसकी स्क्रीन आपके यहाँ से चल रहा है सर वहाँ से तो जो स्क्रीन शेयर हो रही है ना वो स्टॉप हो गई है वहाँ से हो गया क्या हो रहा है okay first we uploaded API file for generation of co-crystal molecule. So API is just active pharmaceutical ingredient. Okay. So second we have to upload co-former molecule also because we required the co-crystal molecule generation with the co-former molecule for the further study of increasement of their pharmacophysical properties and all. So you have to just click on it, upload co-former XYZ file and upload number of co-former as you want. Suppose you want 5 co-former with one drug molecule or as of your choice you can upload. So now I am uploading 2 compounds like 4-amino benzoic acid and suppose I am taking this one and this cytosine. Okay. So I am uploading these 2 files. Okay, after uploading you can see here all the structural information regarding the drug molecule, its element composition which is its coordinates, all you can see here. So after that just you need to click on run generation button for the generation of structure as a complex structure of co-crystal with agomelatine with 4-amino benzoic acid and cytosine compound. Okay. So now you can see here your job is submitted successfully for the structure generation through this first step of our CSP. Okay. Then after that this will take hardly 1 to 2 minutes for completion of this 2 molecule preparation. Then next step is a structural relaxation. So once this step is completed, we can move further for the structural relaxation step. Okay. So you can give number of co-former molecule. Suppose you have given one agomelatine drug and 10 co-former molecule. So you can also generate the 10 co-former drug complex with this co-crystal. So now our job is successfully completed. We generated total 2 co-crystal molecule. Like you can see here, it's all information regarding the cell length, ABC cell length, its symmetry and all. So this is a structure 1. You can visualize in our visualization also. Okay. After generation of molecule, we can visualize its unit cell along with API and along with the co-former molecule also. You can see here, this is a agomelatine drug molecule and this is a co-former 4-amino benzoic acid. Okay, so this is a you can visualize generated compound from here. Then you can also download this crystal information file, CIF file, that generated in the first step of the structure generation of SilicoCSP tool. Just you need to click on download CIF, so your file will be successfully downloaded here, and you can visualize in our inbuilt visualizer, or you can use another software also. Then next step is directly we have to pass out these two structure to structural energy relaxation and also the calculation of the total energy per atom for the ranking of this generated crystal structure. So for that, just you have to no need to upload any file again like XYZ file. First we uploaded the XYZ file already in the first step of structure generation. So now just you need to click on this run relaxation button. So by this button, automatically this generated crystal structure are transferred to structure relaxation step. So automatically they will analyze and they will run the GNN model of AI based algorithm and calculate the their energy per atom, and also rank the co-crystal generated co-crystal according to their stability. Okay, we will see after completion of this step. It will take hardly 2 to 3 minutes. And after that, directly in the MD simulation, we can transfer this 2 molecule directly into a last step of the this crystal structure prediction, and we can calculate the all the energies of the MD simulation behavior of the drug molecule and the co-former molecule. So from that, from starting to end, we are going to check the stability, its structural conformation along with the each atom specific interaction in this the unit cell of the crystal structure. So we will see after the completion of this running. Yes, you can see here our second step is completed, relaxation step is completed with all the details of the again all the details of the symmetry of the crystal structure and all you can see here. Again you can download the relaxed structure of the co-crystal molecule with that you given as an input molecule like agomelatine and 4-amino benzoic acid molecule. So after that you can see below here the energy details of the calculation here available, like first is the agomelatine cysteine, agomelatine with cysteine co-former. It shows the energy minus 6.37 kilocalorie, and then this is the ABC is a dimension length of dimensions of the generated unit cell of the co-crystal molecule. So out of these two you can sort out this compound according to binding energy also. Suppose you can see here out of these two compound, out of these two co-formers cytosine and 4-amino benzoic acid, this agomelatine drug molecule shows stability with cytosine co-former. Okay, so you can say that the first compound, first co-crystal molecule is stable to this energy per atom energy calculation using GNN model of AI. So you can filter out like if suppose you given the 100 co-former molecule with one drug molecule, so you can sort out according to energy of the this energy per atom. Okay. After that you can download the all details of the energy calculation through this icon download CSV file. So you can directly give the all the information in this CSV file. And the last step is our MD simulation of this generated co-crystal molecule of agomelatine and these two conformer. You have to just need again the run MD simulation icon. So MD simulation in the backend it automatically run for these two generated co-crystal molecule, and it will give the all the details about MD simulation. Okay, and lastly it will generate the XRD plot of the generated co-crystal molecule. So after completion we will see the result and all. Okay, so meanwhile we will go for the another tool. Okay, the CSP is running MD. We will see after completion. Okay, so our next tool is a sketcher. In our pharmaceutical research area or any for the educational background, any researcher required starting from the 2D structure of the molecule or any type of the molecule required to its structural information details, and along with the conversion of the 2D structure into 3D structure SDF file and all. So this sketcher is very useful for drawing of any type of the compound that available from your side. You can download, you can use many of the icons here, like suppose this is a icon of the any type of the heterocyclic ring, or suppose this benzene ring, you can directly click on it and directly paste it on the this screen. So you can directly draw your molecule easily. Then you can add the bonds also, like double bonds, triple bonds, according to your structure you can add bonds also. Okay, so after that you can add it contains so many elements also. You can directly click on the any element like oxygen, chlorine. Okay, just chlorine, and just click on the point of attachment where you need to attach the chlorine molecule. So you can draw any molecule by this way, and if suppose you draw some molecule and it behaves as a bad molecule, like suppose you given the wrong valency of compound, like nitrogen has the 3 valency, suppose you given the 4 bonds there, so it will detect the structural information as the wrong structure you drawn. So we can fetch the feature also available regarding the checking the structure. The check structure is also available. If you click on the check structure, it will automatically check the structure and it will give the error here, listed out here, what error comes and what error available in your structure. Okay, then you can directly fetch your structure through smileys also. So you have to go this icon, input molecule, open. Okay, so paste from clipboard. You have to first copy the smile of any compound that you want to draw here. Okay, so go to any smile. Okay, suppose I have this SMI file containing some molecules. Yes, suppose these are all the molecule. You have to just copy. You want to draw this molecule A15. Just you want to you need to copy it. Okay, and after copying, you have to just paste on this clipboard. Control V and add to canvas. So you can see here your structure directly pasted in the sketcher tool. Okay. Then after that you can remove your this structure through eraser is also available for the removing of this tool. And lastly you can save your structure into 3D SDF file also from here. So there is a output save as option is also available. So you can go to a save as structure. You can select the any file format if you want. Mold file, if you want SDF file, so just click on the SDF file and you need to click on save. So your SDF file will save automatically in your download folder. So this is about the sketcher tool. It contains along with that also zoom in and zoom out option also there. And this is all about sketcher. Now we will see about our CSP completed or not. Yes, our CSP tool is completed with MD simulation, third step. Okay, so in CSP you can see here the energy at 0 picosecond and energy as 1 picosecond, means energy at first frame of MD simulation to last frame of MD simulation, it is available here. So you can also just fix out this compound by clicking this arrow. Okay, smaller difference between them is good for the stability of this co-crystal molecule. So you can just check the difference from here and arrange the difference using this. Okay, so out of these two molecules, according to MD, this relaxed agomelatine 4-amino benzoic acid is a good compound. Okay, then you can generate the PXRD plot of from this icon. So first you have to click on this. First you have to click on this box for the generation of PXRD plot. Just click on it, and after clicking, your PXRD plot regarding co-crystal is directly generated. Okay. And lastly, after the generation of PXRD plot, you can analyze, interpret your PXRD plot and download. You can download also the PXRD plot for your research purpose or any further formulation in your industry level also. Then after that, meanwhile we seen the sketcher tool. After that, next tool is our MolCon tool. Okay, MolCon is a tool that required for the conversion of one file format to another. I think you are all not aware about. There are so many types of file format available in our field of this chemoinformatics and all, like SDF, MOL, MOL2, then SMI file. Every structure has a SMILE format, INTISKey SMILE format. So in MolCon tool, we added all the feature. If you want SMI file to convert SDF file, you need to just upload the SMI file in that particular tool. It will directly convert into the SDF file. Okay. Okay. Now our CSP's XRD plot has been generated. You can see these both plots are generated. If you want to download, you just click on this relax agomelatine with this co-format. This graph automatically generated. So this is the overall CSP tool demonstration regarding starting to structural generation of API molecule with co-crystal molecule. Okay. Now we are moving for mole conversion. Okay. This is a tool that is required for conversion of the file formats one to another. Okay, so this is the interface of MolCon tool. Here, same, you have to first create a job ID. Whatever job ID you have to first create a job ID for the further execution and all. Just you have to type something. Just click on it. 1 minute. Okay, just you have to click on it, create an job ID. Okay, you can see here your job ID is created. Now you have to drop, suppose you have to convert SMI file into SDF file. You have suppose SMI file format containing number of SMILES of the compound, and you have to convert SDF for the further execution, like molecular docking required SDF file or something.
I'm talking, he is talking about the molecular conversion. So, this tool actually, suppose you have 10,000 molecules. Before that, I would like to ask the crowd, how many of you have done docking? I know some people from RDM here, and okay. So suppose you have docking 1 molecule, 2 molecules, 10 molecules, 20 molecules, 100 molecules, 200 molecules, 500 molecules manageable to convert one by one. Okay, if you have a lot of dedication, you have patience, you can do 1,000. Okay, but suppose you have suppose 20 lakhs molecule. How can you convert? Because what is the conversion of the molecule? So suppose in case of the Microsoft Word, there is some extension DocX, PPTX for PowerPoint, XLX for Excel sheet. Similarly, each of the molecule has some file format in a different file. STF, structural data, what you call STF file format. So like that, mol 2, mol, SMI, that is Smiley's format. So these are different file formatters there. So different tools require the different file format. Suppose in Autodoc tool, Autodoc Vina, they accept PDBQT file format. So Schrodinger, they ask for SDF or MAE file format. So to interconvert this file format, it's very difficult task. So suppose OpenBabel is there, that is also good tool. But if in Windows, if you upload maybe 2,000, 3,000 molecules, it will hang. Okay, and also in my experience, if you go to Linux also, so if you upload 50,000 molecules at a time, it will shut down, it will terminate the segmented fault. So those things is very difficult. That's the beauty here, this tool. This is a very small tool, but it is a powerful tool. So suppose you have few millions of compounds. So then just upload it here, choose which file format you want, and it automatically converts. So during the conversion, the 2-3 things they will do. One is, that is, it will check the duplicacy. Suppose your molecule, the file contains the duplicate molecule, it will remove. If the conjugate molecule is there, it will remove. If any ions is there, it will remove. If any valency issue is there, it will also fix. So in a very short time, the large number of data, it can convert whatever you want to add, hydrogen, or delete hydrogen, or want to add charge, etc. So that is the beauty of this tool. Okay. So before he actually continue, I would like to say that what is the job ID? When is getting job ID? Why need this job ID? So suppose today I am sitting and I am working on this my job, and after that I forget that what I have done, where is my data. So this job ID, suppose after 2 months or 1 month or 1 week you come back and you start the work and want to continue the work. So using that job ID, you can fetch the data from the server. So it will come on your email also, but this job ID is very much important. So you will have this job ID you can get from the email ID also, or here it will suggest what are the previous 10 jobs you had. So by that you can actually do. So these are the overall thing. Actually, I continue.
Okay. So after this upload and conversion, suppose we have to convert SMI file into SDF file. So you need to just click on drop file here, input file, and go to particular folder where you have the SMI file for the conversion. Okay. And here you can see there is folder of molcon. So you just click on, this is a SMI file format containing number of files of compound. Okay, just open it. Yes, I will show you how many compounds are available in that particular input file. It contains total 200 compounds. So suppose you have to convert this 200 compound. This is a SMI file that required to convert into SDF if user want. So this is a smile format. You have to need to give the smile plus the name of the compound. So you have to prepare this type of file for the inputs. So I given this file to molcon of SilicoXplore. So you can see here this file containing 200 compound with SMI. After that, you have to give some parameters of that required for the addition of this file. Suppose your compound you have to need add hydrogen, you just click on it, add hydrogen. If you want to delete hydrogen, you can click on it. As per your requirement, you can click on it. Then suppose you want to generate your compound into 3D SDF or 3D mol2 file or something, just click on the generate 3D. If you want suppose 2D, so you can just click the 2D icon. After setting this parameter, structural modification and geometry properties, you have to just require to select output file. I given this SMI file as an input. I required SDF file. So I have to click on the select output and select SDF from this list of the conversion compounds file format. Then after that, just click on the convert file. Okay. So after clicking this convert file, within short period of time, this 200 molecule will convert into SMI to SDF file format. You can see here our conversion from SMI to SDF is successfully completed. So this is a file. You want to download this file, just click the download button here. So your SDF file will directly automatically downloaded. Okay. So you can see here .sdf file is generated for 200 molecule. So like this way you can generate different file format. Just you need to upload here. If you have SDF file, you want to convert mol to file, just drop here your input file and select the output file from here. Okay. So this is about molcon tool. Okay. So our next tool is molecular docking.
So now we are going to start a docking. That is, the docking is a nowadays the protocol of the drug discovery. You are talking about the computer drug designing. The first thing coming in mind, docking. But docking is not only the drug discovery field, but that is widely used, mostly used. So docking actually help to skin out the molecule. So what is docking actually? So docking is the association between the two different molecules. So when two molecules come close to each other and they produce certain effect. So suppose you have the protein and you have the small molecule. When small molecule bind to the active site, it produces, I think don't take it. Okay. So when it is bind to the active site, it produces certain activity, and that actually helps to treat a disease or control the disease. So two different molecules in a different position they are there will to bring to each other and fixed at the active site. So that is called the molecular docking. So molecular docking may be the small molecule, macromolecule protein, or protein-protein docking, or protein DNA docking, protein RNA docking, small molecule DNA docking, small molecule RNA docking. Also nanoparticle also you can do docking for the drug delivery, and this is another story. So here we will show you how it is easily we can do the multiple docking in a very user-friendly manner. So most of the people have to do, and most of the tools actually like Schrodinger, cassette and everything, you can take one protein or multiple ligand at a time to dock. But here what you have done, so suppose you have 10 proteins in your target. You don't know exactly which protein is good for my for my molecule. So 10 protein you upload it here, upload whatever the number of molecules, prepare it, submit it, one by one all will go. So this tool is one-to-one docking, that is one protein, one molecule, one-to-many docking, one-to-many docking, many-to-many docking, many protein, many molecules. So that actually we will show. So in molecular docking, whoever done docking, there are 5 steps are there. The first step is the protein preparation. We have to prepare the protein to remove the unwanted thing and repair it. Second, the ligand preparation. Third is the binding site identification. It is called the grid generation. And the fourth one is the docking, and the final one is the analysis. So all these things actually we do, we will set up it here and submit it. So yeah, Vikram will explain.
Okay. So in this docking tool, ligand lock tool, you have to first generate same job ID here. So just you need to click on the this job ID, create job ID icon. So you can see here your job ID successfully created. You can also fetch your this whole execution after the some time with this same job ID. So you need to note down this job ID for further checking. Then after that, it contains total 4 to 5 steps. First is the protein preparation. Second is the ligand molecule preparation that we want to dock in the active site of the protein molecule. Okay, and next is the submission of the docking molecule along with detection of the active site or pocket of the protein molecule. So this is the third step. In this we have to generate the grid dimension and grid config file of this protein molecule. So in the first protein preparation test step, you have to upload protein file. Just you have to click here. Okay, just upload protein file. Yes, sir. Then after that, you have to go the folder where you save your protein molecule. Just go and you can see here 2JIU PDB ID file is protein there. You just click on it. And you can see here your protein is successfully uploaded here. So this is the visualization of protein. You can see here it contains 2 chains. Okay, these are the 2 chains and all structural information of this protein. You can see here like chain A containing all the amino acid residue with all details, and chain B with all details of amino acid. You can change the representation of this protein also here, representation and all features. Then after that, for the preparation of protein, you required you required only specific part of the protein for the docking molecule. So for the deletion of unwanted compound, like it contain A and B chain, if suppose you require only A chain of protein, so for the deletion of unwanted part of the protein, you have to click on this setting icon, this. Okay. Then after that, you can see here all the structural information of this protein. You can see here it contain AB chain along with ligand that is attached with this protein molecule. Suppose I want to dock this chain A with 304 amino acid, and I want to delete this chain. So you can click the chain B for a deletion, and also delete the ligand molecule available in that protein, and it contains some water molecule, so you can also delete this also. So unwanted part you can delete just by clicking on this box of the unwanted compound and just save the selection. Okay. Then after that, all the process completed, like deletion of unwanted compound in the protein and all. Answer? Okay. Then after that, if suppose you want to dock some compound with another protein, so you can directly go in the upload protein file and upload the another protein. So here we are doing docking with 2 protein with multiple ligand molecule. Okay, so you can go to this another file 31L PDB-ID. Okay, now you can visualize this protein. You have to hide first protein from visualizer, and you can see here this is the second protein that we uploaded. Okay, it also contains the so many chains. Okay, so same process we have to do with say this protein also, like deletion of unwanted part of this protein. So click on this setting icon, and it contains so many chains. If you want only A chain, so you just click if you select all chain molecule and just remove chain A, means you select all the unwanted chain like B, C, D, E, F and all, and also all the heteroatoms available, and also the water molecule. Okay, and save the changes. Okay.
So here, one thing, the first protein we prepared without, we remove all the, fine, it's fine. Okay, all the protein, all the unwanted thing, that is the co-crystal ligand, water, everything. And the second protein, actually we will keep the co-crystal molecule. Why actually I am keeping co-crystal molecule, I will explain. So just go to settings again, second one. Select second one, settings. So here I will keeping the chain A. So these are the no ligand, am I right? Heme is there, and sodium, guap. I think Cl, yes.
Hemoglobin.
Cl, yeah, the Cl, yeah, that is a ligand, I think so, yeah. Okay. So I want to keep this Cl co-crystal ligand with the chain A and just save it. Now the 2 protein, select both protein. Then now the 2 protein, I actually selected what are the things I would like to keep. And another thing is that in the protein, when you extract the protein from the protein data bank, there are a lot of problems are there, the missing residues, chain break, so that actually automatically you can repair here. So just prepare it. So in this way, actually we are showing here two proteins. You can upload 100 molecules at a time, 100 protein at a time. So it will take some time and then it will prepare. So now the both proteins are prepared right now. Okay? Go to the next ligand.
Okay. So here in the second step of ligand preparation, we require to upload ligand file that we want to dock with these two proteins. Okay. So go to the particular directory of docking. Okay. And you have to upload SDF file containing all the molecules. Suppose you have 10 molecule, 20 molecule, 100 molecule in. So just you need to prepare combine SDF file and just upload here. Okay. So you can see here the 3D structure of this all the ligand molecule you can see in this visualizer. Okay. So here,
yeah, 1 minute. Here in a single file you may have the multiple ligand volume. So in a single ligand the multiple molecules may have, and you can upload the multiple files also. So in this file total 10 molecules is there. You can see that 2 of 10 is written there. So just navigate the arrow. Navigate the arrow. Yeah. So you can see that these different molecules here. So now you need to prepare the small molecule. Why you need to prepare? Because the small molecule may have the wrong valency or sometimes the conjugate molecule. So that we have to remove. So just select, and also you have to add hydrogens and charges. Select that one, and I just prepare it. So you can upload the multiple files here or single file with a large number of molecules. So 6 molecules prepared in a few seconds. Okay. And now we are going to, for the docking, the first thing we have to identify the grid or the binding site of the both protein. Okay. So go to this. So now you can see the prepared protein here. Select the first protein and select the second one.
Yes, sir. This first, this second. Select.
Okay. Second one.
Selected the second also.
And it is not select check box. The second one, 3, yeah. Now you can see here the active site detection. If you click that one, so you will get a 5 options to identify the pocket. First one, the co-crystal ligand. If the co-crystal ligand is there, it will direct you to find out where your active site is. Or if you don't know, you can find the cavity direction. What are the cavities are there? Or you can go for, you can upload. Suppose your co-crystal ligand is in your local system, you can upload it. That will also help. Or you can use the certain amino acid. That will also help. Or maybe manual input, you can give the coordinate of the your active site. So that's why actually I kept the second protein. Now the first one will go for the cavity detection. And define the box. So here you can see the total, if you go down here, total 28 pockets are there. Now you have to decide which pocket you would like to go for docking. Okay. So you just, if you check it, the position, you select different pockets. Yeah, you can see the different place of your protein where it is, and go down and select the preview. So here the coordinate is there for this particular pocket. Preview, it will show the box here. So suppose you are going for the docking, the second pocket number 2. So go to the pocket 2, select it, go to the preview. So now it is here, and now we are saving these details. So save the config here. So this protein active site definition done. Now the Okay. Now select the second one and unselect the first one. Now the co-crystal ligand, show protein first, and eye icon. Yeah, and here with the first eye icon, just unselect it, hide it, first one. Hide it. An eye icon. Click on the eye icon, first one.
Then it's unselected. Hide it already.
Okay. Now, where is the ligand?
So this one.
Just select the first protein first, and the eye icon. Okay. So in the second protein, select the second protein and select the first one. So you can see this one, the red one, you can see the co-crystal. Just rotate it, co-crystal ligand is there.
Yes.
Let's go look at something is there. So co-crystal ligand is there. So we go select the active site selection. Active site selection. No, not cavity. Close it. Go to the co-crystal ligand, auto detect. Okay. So now,
yes.
Go to preview. So you can see around the co-crystal ligand. Just zoom in. Mouse nahi. So you can see the active site defined around the co-crystal ligand. And now it is save it. So both proteins are now prepared and save the active site. And now we go for docking. Select the both proteins. And we have already selected the ligand. If this ligand contains the 10 molecules. And then just submit the docking. So now one by one, the total how many docking will be here? Total 20. One protein 10 molecules, another protein 10 molecules. So 20 dockings will be done in one by one in the back end. Okay, suppose you have 20 molecules, so that same way we will do. Now during the docking, how many compounds docked you can check it here. So go to the check results. So select the protein, first protein may be the ligand, and fetch the data. So till now 2 molecules docked already. Once it completed, you will notify it, but during the process of the docking, how many compounds are docked, that you can see here. So just show the pose, click on the, okay, here you can see the inside the protein. Now rotate it. So this is the first protein, second molecule. So you can see the second molecule here. And so the binding energy is there. So here the first binding energy, first pose is -9, and first pose, go to the second, go to right, little bit right. This is the first pose of 9 poses. So you can navigate second pose.
Second.
This is the second one, that is 8.7. So that way you can check the each pose of the each molecule. Okay. So this way I can again fetch the results. Click on the fetch on the results. So now it is 8 molecules completed. So that way actually it will go. So this way now we are not, because due to time actually we are not going further. So once you have the huge number of molecules, a huge number of protein, so you docked it, and then the post docking analysis on option is there. Just you select that what is the threshold of your binding energy. So suppose I want to see the molecules which having the binding energy less than -8. So just select it, all the molecules will come to you. You just select it and you can see the binding energy. So here go to the again go to the fetch the results, previous one. Show the results and show the binding energy. So here also you can see the binding interaction. So quickly we see the binding interaction. So the last option I think so. Select it, last option. Yeah. So you can see the first one molecule, this is the binding interaction data, and here it is showing that the hydrophobic interaction with the phenyl, alanine 723, alanine 743, like that, hydrogen bond with this amino acid, pi S this one. So this way individually you can select, or you can download this CSV file, that is suppose in your paper you would like to give the table, so you just download the table and just paste it here. So this is all about the docking. So the thing is that this tool will already remove the barrier of your limitations. Your limitations is like computational power. You need the Linux environment because it's a Windows is not possible to handle such data. So the Linux barrier also to screen out the molecules. You need the programming language like Python is very much useful. So that also remove. So that is the beauty of this tool. Okay. So now we conclude this docking tool here. So we will go which maybe DFT will show very quickly.
Yes.
So you will see. We will come. We'll later. We'll see if time permits. We'll do. Go to the preview. Okay. Now we'll actually quickly show you the density functional theory tool. So homo-lumo energy calculation and the energy barrier you can see. So this approach is computationally very much expensive. So if you have a standard molecule around 20 or 30 atoms, so it may take a few hours to maybe 1 or 2 days. So here actually we will see very small molecule, maybe one phenol or something like that. एक phenol draw करो ना.
सर स्माइल्स कॉपी कर लेना.
Okay. So here actually you can draw your molecule, create the job ID first, and then okay. Better delete it.
Yes, sir.
Delete the smileys. Draw here. Take the benzene and just add the chlorine or hydroxyl group.
Okay.
एक chlorine डाल दो, chloro.
Okay.
So suppose we are going to calculate the homo-lumo energy of this one. So just and then just click on the run option. So maybe I think 1 or 2 minutes it will calculate. But if you have the bigger molecule, it will take much time. Actually, sometimes it will take 2 or 3 days also, because the computation is very much expensive.
Apart from that, you have to select here functional basic set and required for the DFT calculation. So there are some common sets are available in DFT study. So you have to select here the functional and basic set here like B3, LYP, 631. So we provided I think 4 to 5 basic set also. So you can select any according to your research purpose. And after that you can calculate this whole like geometry optimization, Mulliken charges, dipole moment, homo-lumo energy calculation of particular structure molecule, along with electrostatic polarization of electrons, overall map of ESP map, and also density available in the electrons in the molecule. Okay, so you can calculate this whole properties here in the DFT. Now you can see here our job is running with generating ESP map and the density surfaces. Okay, so after completion, all the calculation you can see below this platform in the result section. Okay, so it will take hardly short time, means 2 to 3 minutes for small compounds like benzene, phenol, toluene, or any molecule. If you want to submit for drug molecule, so it will take according to the molecular structure and all, hardly it will take 15 to 20 minutes for the calculation of whole the electronic level calculation in the DFT. Okay. So we can study whole ground level of the atomic level of the energy and all elemental availability of whole structure of the molecule. So this DFT is very helpful for it. Okay.
After that, just show the binding site identification, and that then Kunal will show 1 or 2 tools.
Yes. Yes, okay.
Any question for this? Apart from that, what is the price, or it is freely available or not? Apart from that question, if you have any other question, I'll have
Regarding docking, sketcher, Molcon tool, and CSP tool, any question, any doubt? If you want, please ask. Sir, 2 to 3 minutes. ठीक है, तब तक we can start another. पता नहीं सर.
So in between, we will go for another quick tool, that is the binding site identification, because most of the people, when they do docking, it is very much confusing. That is which pocket I should take, and I know the cavities are there, which cavity I should take. So that actually this tool will help to find the appropriate. Okay, so here just upload on protein quickly. So we uploaded here this protein, and we would like to find out what the pockets are there. So find pockets.
लिंक है पूरे बोर्ड से बोर्ड में बैठे बैठे एक सेंटर पे देखते
So you can see here all the how many pockets you can see. These are the pockets are there. So select all the pockets. Pocket 5 pockets or 10 pockets. Okay. So here actually the position of the different colors of the pocket, and if you go to the here, go down, right hand side, go down. So here the pocket details are there. So here the first pocket showing the what is the pocket score, drug score, alpha sphere, MC volume. So these are the different parameters will help you actually which cavity is important for your molecule. So that actually help. And go to the compare the pocket. Left side down. Compare the pocket. Here this is the in a plot that will also actually help you based on the drug score or pocket volume and comparative analysis among the pocket on in your site. Then you can select the pocket. Okay, that's all. I think, yeah. So these actually this is a complete package which actually guide you step by step to select your better molecule and better parameters. Okay, DFT completed?
Yes.
Okay. So now the DFT completed. This is the optimized structure here. I think there's a bond is the problem with the chlorine and that thing I think. So the go down. Very. So this is a chemical reactivity descriptors, all these things. And where is the homolume plot?
Yes.
Yeah. So this is the homolume plot. We are not going to discuss all these things here. This is the chemical reactivity actually assessment, all these things. So the LOMO. This is the LUMO actually, ESP. So these are actually the electronic distribution, how in your molecule electrons are distributed, that actually can show electron density. This will actually help, these are the things will help you to optimize your molecule. Okay. So now we actually step here. Now we will go for few AI based tools, and then okay. Yeah, come. So total 26 tools are there. It is not possible of single day to demonstrate all these things. Few were showing here. So the docking, DFT, and all these things. Now we show you some machine learning based tools.
So now I'm going to show some AI and bioinfo based tools which help in drug discovery pipeline and speed up the whole process. So we'll start with first one. Okay, semantic search. So semantic search is a tool used to find a compound similar to the compound which are active against certain diseases. So it's based on a principle. If some compounds have similar structure, they tend to have similar activity against the disease target. So we'll start with making one job ID. So now this is a fingerprint. Now what's the fingerprint? So fingerprint is we convert the molecular structure into the fingerprint format like binary vectors. Now if you don't
Now,
know,
1 minute.
Yeah.
So what is the purpose of this tool? Very briefly, because you don't have much time to go details about it. Suppose you have some standard molecule with you. You have some drug molecule, and you want to get similar molecule to that standard molecule, because the similar structure may have the similar properties. So you just upload the standard molecule here, upload your database, and from the database you can narrow down the active similar molecule. Yeah, go ahead.
so the first Morgan descriptors, so binary vectors. In simple words, it's like a phone number, but in the 1 and 0. So for if the some property or like hydrogen bond donor acceptors or hydrophobic studies present in the molecule is get 1, otherwise get 0. So like that, all compounds are converted in binary vectors and compared. So I'll upload the query file first.
The query means that is your active molecules or drug molecules for which you want to similar molecule.
Yes, 1 minute. Okay. So this is query file for which you are finding the compounds, similar compounds, and this is a target file.
Database, larger number of molecules.
Yeah. So we'll select it, and now we set the threshold. Let's get for 6.
0.6 means I want the molecule which have the 60% similarity. You can increase, you can decrease. Okay.
Okay, it's done. So we'll download all results. So you can say we find 2 identical compounds, and some have similarity around 0.93 and 8.94.
Total how much compounds you got? Go down.
Total compounds are
yeah, in a few seconds you got 425 similar molecules. So this actually this data, if you reduce the threshold with the 0.6 we have given, so these are the molecules similar to 0.60% or above. So if you go down, the molecule large. So it is actually, I think, how many in the query database is there?
around I think 88.
88.
Yeah.
In the target database.
In target database, and in query it's more like 32.
Okay. So this way, suppose if you upload millions of molecules, maybe half an hour will take.
Yeah.
So this is so fast actually to get the similar structure.
We tried for 20,000 molecules. It took around 3 minutes only.
So these are the molecules. Then you can take these molecules for further study.
Yeah.
Anything else in this?
Yeah, we can use for repurposing, and we can say for initial screening of the molecules. So let's say if any user like don't have many, we can say you can say like bharosa in it results. So you can go for other thing. That is silico screen. So issue with the like tool like sim search. No, it's a drug molecule. Yeah. For
the concept is that, so suppose you are working on the breast cancer, hormone breast cancer, you have traloxifene, tormepine. So these are the standard, already standard molecule. So you want to get similar kind of molecule. So you just input, give those molecules, and you take any database, zinc database, PubChem database, Campbell database, whatever the database. So from those database, it will search the similar molecule of your input, those raloxifene or tormepine molecule, and it will bring to you. So it will give. Now it is no need of receptor right now, because you already know these molecules for the breast cancer.
Yeah.
It is established. And then you can go for these molecule docking and everything else. It's a different story.
Like even after we getting this compound for initial screening, Problem with a tool like this, not every time similar compounds or similar activities. If you add one methyl group in it, potential totally changes. And one more thing, some compound, these tools don't like most of RDK based or Morgan based tool don't consider the chirality for enantiomers like dextromethamphetamine. If you hear about it, it's a strong CNS excitor, but it's a Leo version. It's used for like in the inhale errors. So it's totally different activity for same compound. So after getting this, we can go for SilicoScreen. Now SilicoScreen is a special one. So what SilicoScreen does, it's a virtual screening tool. It's based on the AI-based virtual screening tool. It takes compounds and like produce active, checks if active or inactive. But basically, it's a compound active or inactive. So first,
here actually what we have done here. Suppose I want to work with protease, HIV protease. So what I have done, I have collected the HIV protease active molecules or inactive molecules. So we know these are the active, these are inactive. And now I use the machine learning to develop the model. So what are the features made them active, what are the features made them inactive. So that concept I have given to the model. So the now model have the features, these patterns should present in the active molecule, this pattern should present in the inactive molecule. So once this model I have plenty of molecules, I don't know whether it is the protease inhibitors or not. So I will fit those molecules in this model and it will identify whether this is active for protease or inactive for protease. So based on the 0 and 1, that is actually the concept.
So basically, if you see the data, just 1 minute. So you can see this is our smiles for HIV, and one is a label. One means it's active for that disease. Means how is it have ability against this one, means active against it. So we prepare a data set like this with the compounds active and inactive. So now why we comparing to active and inactive units, I will show you. What we do, we use this data for the training. So batch size is a number of molecules model sees during a training. Learning rate is a activity.
So he is actually developing the model right now. It is not the pre-developed model. The machine learning model he is developing here only. So he has the active molecule, he has the inactive molecule, giving the input and the process.
Epoch is a training cycle basically, and dropout is used for like to get better result, because model tend to get like neural network models overfits. So we drop out some layers out of it. So this tool is basically used for initial screening. We can take it's a disease specific. We can train the model for a particular disease and screen the thousands of compound at once. So we can get.
In this tool, the option is there. If you have your own data, you trained your model here only. And if you don't have your data, so some pre-developed models are there. So you can use those for your target.
Okay, it's done. So you can analyze the results. So this is a validation loss and training loss. This is AI thing. We can see the loss means the model a number of errors model making during training. So it's continuously decreasing, and validation is going to decreasing. Okay.
So these are the quality. Actually, I am not going to take that because of lack of time. So these are the quality will tell you. Then you can retrain your model. Suppose you are not satisfied with the data, statistical data, accuracy is not good, efficiency is not good. You retrain the model.
And this is why we are using it both active and inactive, because we need the data set to be a balanced one. Because if we use data set which is one-sided, so model will be biased with total results. So now we predict activity.
Now actually once it done, now we will predict my unknown data. I don't know this data contains active or not inactive molecule. So that actually we will use here to predict.
So the threshold set to be 0.5. It's a basic one. If the molecule is a probability error 0.5 means it's active. Below that is inactive. So this is probability.
So here you can see, that is, if you see the left hand side, 1 means it is predicted active. If it is 0, it is inactive.
Inactive, yeah.
So you can navigate next I think 128 molecule like that. So then here you can actually, you will get idea, yes, these are the molecule might be active, but you need the further confirmation, other tools, and finally the experiment evaluation. But this will help you narrow down you, yes, this possibility is there is active or inactive.
Yeah.
Okay, next, go to the next tool.
So now we got hits for the particular target. So now what we're gonna do? We go for QSAR.
So QSR is actually the quantitative structure activity relationship tool. It will help to predict the biological activity before going to the experimental calculation. So you can develop your QSR model here and use that. You can check, yes, what possibly the biological activity will be there in nanomolar level, micromolar level, or what level. So that actually you can predict here.
So we're using this data for training and this data for predictions. So what we do is here QSAR model is a from name you can understand is association relation, makes association relationship from descriptors it predicts the activity. Now what's a descriptor? Descriptor are just numerical representation of a molecular features. So we can say 2D, 3D, or both of them.
2D first turn, na?
Yeah, 2D.
Yeah, just only select 2D.
2D. And what is the feature selection? Models generate around 200 features. Not all of them.
Regressor even.
Yeah. Important for the their activity. So we just scale them and remove the.
These are the different parameters. Actually, you have to decide what parameter I should take. So these are all actually user specific. मुझे कौन सा लेना है, मेरा डेटा के लिए कौन सा सही रहेगा, that I should have the knowledge.
Yeah. Now this is algorithms. You can select any of them, or you can go for stacking. Stacking means you can multiple algorithm at once.
Random forest actually is stacking here.
Yeah. So next is selecting activity conversion. So IC50, what's IC50? IC50 is a concentration compound required to inhibit the 50% of biological activity. So PIC50 is just a negative algorithm of it. Now I will run the predictions.
So these are the tool actually enormously will help you speed up your research actually. So that is an easy way. You have to do nothing.
Now you may have question, if we have silico screen, why we are using this QSAR thing. So silico screen only give us the active or inactive, but QSAR gives us the how active is the compound is with the predicting a PIC50 value. So you can go for finding it with the silico screen and then go for lead with this compound, selecting the best out of them using predicted PIC50. Yeah. So this graph is we can I explain it in easy words. So this is it's called a line of fit in our language. Means these are the predictions. If the predictions are closer to line, which model making correct prediction on the unseen data, means the test data. And this is we can say a correlation graph, predicted value and actual. So it's lie close to the line, so prediction are mostly correct. So this is a predicted PIC50 value 5 and IC50. The lower the IC50 value, it's better the molecule is. So confidence is around 0.98. So it's good.
So this actually gives you some idea that what kind of molecule is, what is the possibility to be active or inactive molecule. Okay,
so
next.
Number 616 is the best out of it. It have only 16 IC50 in nanometer. So we can use it for further we can say screening. So this is out. Okay.
Next one.
Next is toxi.
Yeah. So toxi, the toxicity is a very serious concern. So before going for the experiment, we need to calculate that is molecules are toxic in nature or non-toxic in nature. So that actually easily you can predict here.
So
and this is also AI based model.
Yeah. Toxi is a pre-trained model. Okay. So issue is that many molecules pass all the tests and only to get failed at the clinical stages. So we need to remove such molecules early on. We can we have two options. You can use smiles here, or you can draw here, or you can give all smiles containing file here. So we go with this one option. and then there is toxia. So this one, okay. So this is threshold you can set. So toxic threshold you can change it, because for cancer drugs you can have little toxic compounds, but for regulated drugs you can have this one. So we can set the threshold from here, or you you can go with the default toxicity above 0.65. So toxia give results in 0 to 1. So if the result is 0.61 above, so it's toxic, and it's between 0.15 and 65, it's a medium one, or 0. Below that is like non-toxic compound. So we'll make the predictions now. So one more thing, it's a generalized toxicity prediction model. It's not for particular toxicity like hepatotoxicity or anything.
Specific actually, it is overall. Tissue specific toxicity tools actually are under development. Will come on that very soon.
So this is all toxicity result we got. So this compound, it have very low toxicity in minor scale, and the synthetic toxicity is the thing how easy to make the compound in the lab, wet lab. So lower the value is, better it is. So easier to make the compound in wet lab. So it's 10 for it is like not very good. Now Lipinski rule of 5. It's says how orally available the molecule is. Okay. Yeah.
So these are the physical chemical properties that actually help to decide the synthetic feasibility, Lipinski rule, all this etc. Surface area. So these actually help actually. Go to the tool.
Yeah.
Go to tool. So here actually the graph, it is showing that total input I think 319, 110, 429 and 5. Around 1000 molecules were there. We checked the toxic and non-toxic. So we found 5721 molecule toxic, 110 non-toxic, and 319 that is in a borderline. That is, you can consider, but very cautiously. So that actually help you if you have a large number of molecules. What are the molecule I should go, and then you can download from here actually.
Yeah, you can only download toxic one, moderate one, or simple one too.
What tools you have?
Yes, end. It's end. Khatam ho gaya.
Okay.
Yeah. So you can also draw it with using that any compound. Let's say
for the this is for single molecule.
Yeah.
But for the large number of molecule, you can save in the Ismailis form.
Yeah, you can go with pyrimidine. Okay, it must be toxic. Okay, yeah, it's toxic to inhale for humans, so it come out to be toxic.
Okay, now go to the main home page.
Home page, yeah.
So this is actually very harshly. We have completed few tools demonstration. जब हम बहुत जल्दबाजी करते हैं तो उसमें मज़ा नहीं आता, तो हम कोशिश तो किया है, लेकिन जो दिस इज अमेजिंग टूल एंड जो है ना बहुत सारे टूल्स है, जैसे मेटल डॉकिंग. दिस इज excellent tool for the metal docking, because metal docking is a very difficult task in a normally, but we made it easy here. Then molecular dynamic simulation, this is very tedious job. People डरते हैं, लोग डरते हैं कि कैसे करना है, because this is so क्या कहते हैं यूजर अनफ्रेंडली टूल, हर बात पे एरर देते हैं वो. तो उसको जो है ना हमने सॉल्व किया कि जैसे आपको कुछ करना ही नहीं है, आप मॉलिक्यूल ले आओ, अपलोड करो, रेस्ट विल बी डन इन द बैक एंड एंड यू विल गेट द रिजल्ट्स. देन या फार्माकोकाइनेटिक्स इज देयर.
इस फॉर्म क्या है?
फॉर्म क्या है? जस्ट सो वेरी क्विकली. सो या जस्ट क्विकली सो ऑन डेटा.
फॉर्म क्या है?
यस. Now it is actually the we are categorizing like Sutex, ecology and all these things. So we are categorizing right now. It is overall right now. Maybe in a few months we will categorize for the different area. How can it be used? So pharmacokinetics is there. So he is just calculating the pharmacokinetics parameters here. अभी मत करिए, 2 मिनट के रुक रहिए फिर. So apart from that, actually, sometimes we are train the people. That is, if you want to learn some skill development, that also will provide. So we provide. Already I mentioned that is, we will provide the internship to students. So depending upon our requirement, students' requirement, and then we discuss and then we provide. So anyone interested for internship, or maybe also sometimes we advertise for job. So if you have the skill, then definitely you approach to us. But one thing at the end I should say, I must say, that is, we want to survive in this world, in this time, so please learn Python and machine learning. Otherwise, you have to, the thing is that right now, maybe in a couple of years, it will come like that. To save my job, I have to learn Python and machine learning and artificial intelligence. Otherwise, I lose my job. That is the situation is coming. So please, it is the time to the students. Please give extra time, learn Python, learn machine learning. It is not the big deal. If you just invest your 1 or 2 hours per day, a lot of free tools are there, a lot of free videos are there, a lot of tutorials are there. Just go through. So if you just manage some time from the Insta to Python, so I think your future will be secure. So that is actually my message, because I can understand when I was in academic, I had the different feelings about the market, but when I came to the corporate, totally different. Why I should hire a person who don't know the current skill? Why should? Because the company is running based on the project done by the employee. So most of the companies, they don't want to invest 6 months or 7 months to invest the student. They want, that is, yes, if I hire someone, within maybe 1 month or 2 months, the candidate should be ready for the work. So that skill, if you actually develop from now onwards, then you will be ahead in the job interview. Okay, so here you can see the all the molecules done. So if you select on the molecule, you click on the absorption. So in the that is category, click on the absorption first. So here it will show you the Kakoevang, it is bad or good. Then Pampa, I don't get the meaning, the distribution. So these are different parameters will calculate and it will tell you the pharmacokinetics, whether this molecule is pharmacokinetically acceptable or not. Go to distribution. These are the here. Then go to the metabolism. So these are the different CIP, T6 and all these parameters. Go down. The pharmacokinetics here. These are the different parameters. Now go down. Physical chemical properties are here. And finally the toxicity parameter also there. Go to toxicity. So these are the here, like MS toxicity, carcinogenicity, and all the molecules you can download. Go to the download page. You can download the CSV. The total go up. Download CSV. So all the 1000 molecules, all the parameters are here. Now we can screen it based on this. So this is just actually just to start it. So now we categorically we are doing. So thank you so much everyone for your patience and your support. And especially I'd like to thank Director Sir, Principal Sir, and all the organizing committee for a wonderful event. Thank you so much.
Thank you. Thank you so much, sir, for your enlightening and insightful session. It was actually a very new experience for us to get a hands-on training on the things we have learned in our books. Thank you so much. Time was very quickly going and we can't get to know more things. Thank you. Now it's a final time for a vote of thanks. It's my proud privilege to propose the vote of thanks on this memorable occasion of the valedictory function. I request our Principal Sir Dr. Vinod Thakre and Professor Dr. Pramod Khedekar Sir to please come on the dais and present a vote of thanks to our to present a token of love. To present a token of appreciation, Dr. Now I like to welcome Vikram Singh Suryavanshi sir to receive his token of appreciation from Nagpur College of Pharmacy. I request Mr. Kunal Saire sir to please come on dais. I request Mrs. Suhani Gawane ma'am to please come on dais to receive his certificate of appreciation. Thank you so much. It was a privilege of Nagpur College of Pharmacy. Now it is my proud privilege to propose a vote of thanks on this memorable occasion of the valedictory function of World International Seminar and Drug Discovery Training Program. The first foremost, I express my heartfelt gratitude to our Chief Patron, respected Chief Patron, Honorable Shri Dattaji Meghe Sir, respected Patron Shri Raviji Meghe Sir, Shri Raghav Meghe Sir, Shri Samirji Meghe Sir and Shrimati Vrindatai Meghe Ma'am, respected Convener, Principal Dr. Vinod Thakre Sir, respected Co-Convener, Professor Dr. Pramod Khedekar Sir, Honorable Speaker, Dr. Amit Pant Sir and Mr. MD Atahul Islam Sir, respected IQAC Head, Dr. Pradeep Vaibhav Uplenchivar Sir, I extend my sincere thanks to our convener, and we are deeply grateful to our esteemed international speaker. My sincere appreciation goes to the organizing chairs, Dr. Surse Sir, Dr. Uplenchivar Sir, Dr. Hiradeve Sir, Mrs. Mane Ma'am, all the coordinators, all the members of organizing committee for their dedicated teamwork and tireless efforts behind the successful execution of today's event. I would like to thank all the committee members responsible for the registration committee, Dr. Lalchand Sir, Madhav Sir and Hina Ma'am, Discipline Committee, Technical and Stage Committee, Mrs. Priya Ma'am and Ms. Shruti Ma'am, Hospitality, Food Committee, Invitation Committee, Campus Visit, Transportation, Feedback, Sujata Ma'am and Renuka Ma'am, and a live session coordination for their sincere and coordinated contributions. Special thanks to our M.Pharm and B.Pharm students who coordinated well for the success of this conference. A special note of thanks to all the faculty members, participants and students who participated and actively engagement added a great value to this seminar. Before I conclude, I express my heartfelt gratitude to everyone who directly or indirectly contributed to making this program a grand success. With this, I humbly concluded the vote of thanks. Thank you for your gracious presence. Have a pleasant evening and a safe journey. I request everyone to please stand for the national anthem. Class stand at ease. Class attention. हाँ सर. भारत माता की वंदे. Thank you so much. Vishram.